untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada Crop Life Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani, agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. Crop Life Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
Food doesn't come from the grocery store. It comes from the land, which means if we want this, we need to protect this and them. How can we produce enough food while also nurturing the land and the farmers who grow on it? 2 words: sustainable agriculture. That means the soil stays healthy, the environment stays healthy, and the farms, you get it. So what does that look like? At Corteva Agriscience, it looks like seeds that are adapted to different environments. So farmers not only conserve much-needed water, they increase the likelihood that seed will make it to harvest. Our biopesticides help farmers control pests and protect crops, naturally. And it's not just our products. We're advising farmers on sustainable farming practices, maximizing yields, and enhancing food quality. Corteva knows that the future of food requires reimagining how we grow it, protecting where we grow it, and ensuring we can keep growing it.
Sustainability. It's a word you hear a lot. But what does it really mean? At Corteva Agriscience, sustainability is more than a word. It's who we are and what we do. We're in it for the good of farmers. We're working hard to increase the livelihoods and sustainable farming practices of 500 million smallholder farmers. We're in it for the good of the land. We're improving nutrient and water stewardship, productivity, and soil health on 30 million hectares of global agricultural land. We're in it for the good of our communities. We're empowering women and youth across the globe to help them become successful business owners and providers for their families. And we're in it for the good of the planet. We're reducing greenhouse gas emissions, conserving water, and enhancing biodiversity in our operations and supply chain. Sustainability. It means more than ever, for us and for the world. It's the foundation of our future, and we're building it right now. For farmers, for consumers, for our planet. Together, we're in it for good.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions, science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
When the genetic code of our DNA was discovered, our world changed. We had identified the biological blueprint that defines everything about who we are. From there, the technology to explore our genome evolved rapidly. Programmable DNA editors like zinc finger nucleases and talents could change specific genes, and RNA interference could prevent gene expression. These tools transformed us from genome explorers to genome engineers. We could not only observe, but also change genes and investigate their impacts to understand how our biological blueprint is read. The advent of CRISPR-Cas9 technology brought versatility, expanding the engineer's toolbox to include virtually any type of change, from gene silencing to custom sequence insertion to transient activation or inhibition. Anything is possible. The efficiency of these methods allows us to explore and engineer on previously unimaginable scales, studying entire genomes and gene expression in single cells. Genome engineering has even entered the clinic, initiating an exciting new era of personalized medicine. CRISPR can generate a very rare gene processes that target and gene therapies to repair damaged genes are being tested to treat diseases. Our team is here to make all your genome engineering ideas possible with expertly optimized tools to create any type of modification at any scale. We provide more than just reagents. Our scientists also partner with you, sharing their knowledge through technical support to help you take those ideas from possibility to reality. The future of genome engineering is filled with the potential to transform medicine, and we look forward to building the foundations of better health with you.
Halo Sobat Merk, apa kabar? Aku Intan dan aku akan membawakan kabar terbaru dari Merk. Merk sebagai prinsipal kini sudah terdaftar dan hadir loh sebagai penyedia dalam e-katalog LKPP. Nah, Sobat Merk bisa menemukan produk kami di etalase nasional untuk katalog peralatan laboratorium dan etalase lokal untuk katalog bahan kimia. Wah, sebuah kabar yang menggembirakan. Benar kan Sobat Merk? Yuk, kunjungi website e-katalog LKPP dan mulai berbelanja sekarang. Tapi gimana ya caranya untuk temukan produk apa saja sih yang ada di e-katalog LKPP? Ingin tahu kan gimana caranya? Stay tune ya. Bye!
CJ Indonesia adalah bagian dari sebuah bisnis global CJ yang berpusat di Korea Selatan. Memulai ekspansi bisnis pertamanya di luar negeri yang berlokasi di Indonesia pada tahun 1988. Kini perusahaan telah tumbuh menjadi korporasi besar, memiliki beragam unit bisnis di antaranya bioteknologi, feed and livestock, food, logistik, bioskop.
Good morning and welcome to all participants and speakers. In our second day of the
Seiring
International
perkembangan
Conference
pasar dan
and Workshop
perubahan
in
tren
conjunction
dunia
with the
meningkatkan
8th
kehidupan
Indonesia Biotechnology Conference 2023, we'd like to remind all the participants and presenters who haven't registered, please register yourself at the registration desk outside of this hall. And we would like also to inform you that the snack and coffee break is available from now. So please enjoy the refreshments that have been provided by the committee and the hotel. And you can also take your snack and coffee here while enjoying the plenary session, as we will start the second day with 5 invited speakers. Thank you.
Dalam pengembangan produk White Bio, CJ Bio kini telah berhasil memproduksi produk bioplastik polihidroksialkanoat atau PHA. PHA adalah salah satu bahan untuk membuat plastik ramah lingkungan. PHA adalah polyester cadangan yang diakumulasi sebagai butiran intraseluler oleh berbagai macam bakteri. Polihidroksialkanoat atau PHA dihasilkan melalui beberapa tahapan diawali dengan pemrosesan bahan baku raw sugar menjadi glukosa. Proses sakarifikasi pencampuran glukosa dengan PHA strain dilanjutkan dengan proses fermentasi yang menghasilkan produksi intraseluler. Setelah itu dilakukan proses pemurnian atau purifikasi, yaitu pelepasan unsur PHA di dalam sel untuk menghasilkan polimer PHA. PHA adalah satu-satunya plastik yang dapat terurai secara hayati 90%. Dan dapat terurai setelah 120 hari di lautan. Sejalan dengan filosofi CJ dalam menjalankan bisnisnya untuk berkontribusi pada perekonomian nasional, CJ akan terus menjadi perusahaan nomor 1 yang bergerak dalam bidang bisnis bioteknologi, yang terus peduli pada kelestarian lingkungan dan maju berkembang bersama Indonesia.
ITS Science Indonesia is one of business division under ITS Group with headquarters located in Singapore. ITS Science Indonesia offers various analytical instruments for laboratory, medical instruments, and laboratory furniture. For more than 40 years, ITS is moving forward with a vision to make an impact in the industry they serve and a mission to serve better. ITS has core values to put professionalism, partnerships, and performance forward. ITS Group is providing its services around Southeast Asia countries such as Malaysia, Singapore, Thailand, Vietnam, Indonesia, Philippines, and Cambodia. In Indonesia, ITS Science has 2 offices, located in Jakarta and Surabaya. Inside ITS Science Indonesia office, there are facilities to support the work such as training room, and demo lab. ITS Science Indonesia also actively create or participate in various events such as webinars, seminars, product training, and exhibitions. Here are some brands that ITS Science Indonesia is distributing. Products that distributed are from top and reputable brands worldwide. We are also proud to serve the needs of our customers, from governments, hospital, industrial, to universities. If you need help for your laboratory needs, contact us, ITS Science Indonesia, bridging science, inspiring possibilities.
Well, ladies and gentlemen, we are about to start our second day, so please make sure if you have friends or colleagues outside, they are still outside of this hall, please contact them, and we kindly ask you to help us to direct them to this hall as we will start our second day with plenary session. Thank you very much. Well, ladies and gentlemen, we cordially invite you to be seated as we will start our second day of the conference. Thank you. Good morning, ladies and gentlemen. Om Swastiastu, Assalamualaikum warahmatullahi wabarakatuh, Shalom, Namo Buddhaya, may all blessings be with us. Well, ladies and gentlemen, welcome again to the International Conference and Workshop in conjunction with the 8th Indonesia Biotechnology Conference here in Prime Plaza Hotel Sanur, Denpasar, Bali, today on the 17th of May 2023. To all of the participants and speakers and distinguished guests, we hope you had good sleep and prepared your presentation very well, as later on in the afternoon we will have parallel session where participants that have submitted their research papers will present their findings, which we believe will be very fruitful for all of us. And today we will begin the conference with plenary session with the theme of biotechnology and biodiversity use. There will be 5 invited speakers presenting their research. We will hear more interesting topics as we have more presenters today than the day before. Therefore, to presenters online and offline, please kindly stay with us for the whole session because we believe we don't want to miss any important information from our invited speakers. And we would like also to remind you that to offline participants present with us today, you may enjoy the refreshments provided by the committee. You may take the snack and coffee to this hall while enjoying the invited speakers presenting their research. Well, ladies and gentlemen, without further ado, we would like to start our second day of the International Conference with plenary session, accompanied by our moderator and also 5 invited speakers. Without further ado, we would like to invite our moderator today. Our moderator today is a researcher in Indonesian Oil Palm Research Institute. She is also the member of IBC, and her research interest is plant molecular biology in sugarcane and oil palm. Therefore, ladies and gentlemen, please give a round of applause to Dr. Hayati. To Dr. Hayati, time is yours.
Okay, thank you for the MC. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Namo Buddhaya, salam sejahtera untuk kita semua. Good morning, all distinguished speakers, distinguished participants, and also all the participant and committee. Today, we will begin the second day of the International Conference and Workshop in conjunction with the 8th Indonesian Biotechnology Conference, IBC 2023. So please allow me, Hayati from IOPRI Bogor, as a moderator for this session. In this third plenary session, we are honored to have 5 invited speakers, all related with the topics of biotechnology and biodiversity use. So I would like to invite to come forward with me. The first one is Professor Eni Sudarmonowati from National Research and Innovation Agency, or BRIN. And then Dr. Rong Di from Rutgers University, USA. Dr. Ines Slamatludin from International Rice Research Institute, or IRI, Indonesia, and also from IBC. Professor Gede Putu Wirawan, PhD from Udayana University, and also from IBC. And Mr. Eilot Lam from PT Hartek. Mr. Eilot Lam is already here? Yes. Please. I'm sorry. A bit more. we are a little bit dense. Okay, ladies and gentlemen, first I would like to introduce the CV of Professor Annie Sudarmonowati. I think most of all already know her. So Prof. Annie is a researcher on Research Center for Genetic Engineering in BRIN, or National Research and Innovation Agency. She's also a visiting professor in Department of Plant Biology, School of Environment and Biological Science, Rutgers University, New Jersey, USA, and a Species Survival Commission, or SSC, member of IUCN, International Union of Conservation for Nature, and Global Trees Campaign, and also the associate editor on International Journal Malaysia Applied Biology. Her education background, she obtained a PhD from School of Biological Sciences, University of Bath, UK in April 1991, and also the BSc from Bogor Agricultural University in 1985. Some honors and award is Satya Lancana Wirakarya from the President of Republic Indonesia, Best Inventor, 100 Best Indonesian Women Researcher from Ministry of Women Empowerment and Children Protection, 113 Best Innovation in Indonesia from Ministry of Research and Technology. So her research interest is genetic improvement, genetic conservation, biodiversity assessment, genetic engineering, marker-assisted selection, cryopreservation, omics, and NGS. So it's very complete from molecular biology aspect. And today, Prof. Annie will present an interesting topic about bioprospecting from plant diversity applying integrated omics approaches. So, Prof. Annie, you will have about 30 minutes for presentation, and we will have a question and answer later on after the fifth speaker's presentation. So, the time is yours. Thank you.
Thank you, moderator. Good morning, everyone. Please allow me to stand over there on the stage. Someone else? Okay. Yes. Okay, that's fine. Okay, I will start. Please, Ibu Hayati, warn me when it's 10 minutes before the presentation so that I can move very quickly to finish. So yeah, this is good morning again. So I hope all you are now fresh and wide awake. Yeah. So I'm going to start with my presentation. So thank you for, the organizing committee, which is also one of myself there as the steering committee and others, and thank you all friends and colleagues. So my title is Bioprospecting of Tropical Plant Biodiversity by Applying Integrated Omics. So some of the slides maybe you've already seen from Ibu Indra yesterday from the Ministry of Environment and Forestry regarding the type of ecosystem, the type of biodiversity in Indonesia. But I will present it in different way. Yeah, because I have slides that I have to skip some of them, not really skip one slide, maybe one word, so I need to move a bit fast. So the outline of my talk will be like this. So tropical biodiversity as compared Indonesian, global as compared to the Indonesian, and then science and technology developments, especially omics technology, bioprospecting and omics research and application. And fourth, strategy and collaboration. That would be responsibility of many stakeholders. And last, way forward challenges and recommendation. So yeah, I'm coming to the part one. Next. So the global one, I mean, yesterday maybe, yeah, Prof. Erna and also Pak Masteria also mentioned that Indonesia is #1 in the world if we combine terrestrial and marine. So you can see there, it's #2 for the terrestrial. So after Brazil, but then if we combine with marine, that would be #1. So on the slide, you can see that we have 74 ecosystem type. And such number of bird, animal, and others as compared to the global population. So I won't go into detail, but then also on the right slide, you can see that not just the biodiversity, but also cultural diversity. Also, not just culture, and you are here, it's been 2 days or maybe more in here for the foreign colleagues. Also food, culinary diversity. Yeah, next. Yeah, so here, it's too small, but so I just underline what I just said, that at least 200 ethnic groups. It's actually 300 ethnic groups. And the number of species of plants as compared to the world. And also mangrove biodiversity. Why mangrove here are also very high in diversity? Because we are maybe the second after United States longest coastline. Yeah, so that's why we have mangrove, and then sago plants is actually the highest in the world for diversity. So that's why we have to study, save, and use at the same time. Next. So this one, back to the type of ecosystem. So there's the terrestrial. You cannot imagine that in Indonesia you can find from the mountainous alpine types ecosystem to savanna. We have savanna in the eastern part of Indonesia. So you cannot imagine that this is Indonesia. You thought it's in Africa or somewhere. Yeah, and then so freshwater and marine in Indonesia, and also the artificial or man-made ecosystem. Next. Yeah, so this is the ecosystem diversity in Indonesia. So so the marine and also the terrestrial, and what are in marine and then what are in terrestrial ecosystem. So there are a lot. I won't go into details. You can see there are types. One I would like to mention that maybe the highest is the. Up to now, the highest is the percentage or ratio of insect of Indonesia to the world. Yeah. Next. Yeah, so this is species diversity. It was LIPI did the work in 2014, and it was updated in 2018, and we're going to update again. So now it's already under BRIN then. Yeah, so this is the marine and terrestrial also. Next. So I mentioned yesterday during when I asked to the speaker, one of the speaker, about the species in this endemism. So for plants, it's more the highest in Kalimantan. Yeah, so for angiosperm, like 23%, while the highest of endemism is in Malakas Island and Papua. And then for reptile in Sulawesi, and then for amphibia and mammals in Papua. Next. Kalau saya ngangguk langsung aja ya. Yeah, this one, yesterday Professor Slavik mentioned about the degradation. So I also have this slide, but it's different style. So habitat degradation is number one, and then over-exploitation and use of species, excessive use of species, climate change, pollution, and then invasive alien species. So I just quoted here based on the recent working group, two of the COP climate change in Glasgow, that the rise of the temperature can give impact, the reduction like 14% of reduction if the temperature increase 1.5 degrees centigrade, for example, and so on. So that would affect the pollinator. So when the pollinator, now it's already the crisis, when the pollinator decrease, then it will give impact to food availability. Sorry, I put the animation so it's a bit long, so I forgot to turn off the one. So move to the part 2, the science and technology development. So especially for the omics technology, for those who are not familiar with this technology, I will go very briefly. Next. Yeah, so for genotype, so genotype and phenotype, for genotype that would include genome and transcriptome, and then for phenotype that would concern with proteome and metabolome. So I won't go into proteomic and lipidomic, but more into genomic, transcriptomic, and metabolomic. Next. Yeah, so why metabolomic is placed on the bottom? Because it's actually the one that influenced by environment. So that will then affect the higher hierarchy. So that's why then metabolomic, and then transcriptomic, and genomic. Yeah, so this one is just how to describe easily. Yeah, so this one just example. I know it's very small. I mean, you cannot read it. It's not visible. It's visible, but it's not readable. Yeah. So this is just a table to summarize those omics technology, the advantage, the challenges, and also some application. Next. So this one also the application of the omics technology in plants and in mammals, and also for the other products, for example, for nano products, and also for food and others. So this one also the curve telling you that the progress from time to time of each omics technology, so the metabolomic, transcriptomic, genomic, proteomic, and others. So this one also, I show you the applications that already implemented in start bioengineering, for example, for food quality and traceability, genomic, and also for the addressing malnutrition study and also a problem, and plus bioinformatics that will strengthen the technology to solve many problems. So yeah, this one just to show you the technology or the method being used. For example, for metabolomic, we use one of them, LCMS and others, and then for the transcriptomic, we use one of them also, qRT-PCR, and for the genomic, genotyping and or metagenomic, we use the next generation sequencing, including whole genome sequence and also the genome-wide sequence. So this technology actually have plus and minus, so you have to choose based on, depending on your purpose. Next. Now I move on to the part three, the bioprospecting and omics research. So that would include the improvement of the plant, or not just plant, creatures, yeah. So, but I'm talking more focusing on plants. So genetically improved plants, so that could be using genetic engineering. Yesterday we talked about GMO also, and also CRISPR, genome editing today. So Dr. Ines and Dr. Rong Di will be talking about that in more details. So I just give the flow chart. And also then we can modify the plants also using chemicals or using stress induction, for example, during their growth or in the culture medium. And then also the type of culture also influence the trait later on that we can, although maybe stable or not, or we need to test, but solid medium or static medium as compared to the liquid medium. And then type of organs, so whether it's leaves, fruit, or root. So I give you that the result did by one of my team when she was in Netherlands, in Wageningen University, to do the metabolomic. This one is comparing chili, pepper around the world. It's more on diversity things as compared to the content of metabolites. Yeah, so this one, why we need to do bioprospecting? Is it now or later? Of course, now is already too late, of course, but it's better late than doing nothing. And we need to think about the strategy. So in the US, for example, pharmaceutical industry, 25% of all prescription of drugs sold, it's actually being derived from plant-based sources. And also of the number of approved drugs, it's over 62% of cancer drugs approved by US FDA of natural origin based on natural products. And if you compare with tropical rainforests, it can be in Latin America, in Amazon forest, or in Indonesia, or in the Asia forest, that of course is more interesting because they provide more diversity in that as compared to the temperate regions. Next. So this slide just to show you successful bioprospecting program. So from drug, antibiotic, cosmetic, and nanotechnology and biosensors, pharmaceuticals, bioremediation, and also functional food. Down on the slide, on the bottom, I underline one antibiotic that still use now, vancomycin, is actually found from Kalimantan. So someone sent the soil, and then they isolated the bacteria, and then it's actually grew Streptomyces orientalis. So that time there was no access and benefit sharing or cost of benefit sharing. So that's actually from Indonesia, from Borneo. Next. So it was since 1950 something. So to do that, we need 4 stages. Yeah, 4 stages. Just summarize here: exploration and expedition, and then inventory, identification, characterization, plus research, screening and research also, and product development and commercialization. It takes time. It takes time. So, and it costs a lot of money. Yeah, for exploration expedition, for example, Indonesia, I mean, last time LIPI, before it's merged into BRIN or transformed into BRIN, we have to collaborate with a lot of foreign researchers, scientists, for example, with the US to explore our Sulawesi forest. Yeah, that time, and using our ship for vehicle, for research vehicle, and then it costs a lot of money also. So each stage actually needs a lot of money, especially for commercialization, because before that you need to do preclinical tests and then clinical tests. So. And also we need to collaborate. We cannot do that. As a scientist or researcher, we cannot do that. We have to collaborate with industry, and then we have to collaborate with hospitals also. Next. Yeah, so this book is not really fresh from the oven. It was last year I published. So this one, as an editor with a professor in Bogor Agriculture University or IPB University, but also it's actually like 16 chapters, and I wrote like 6 or 7 chapters. Yeah, so yeah, this one, why we need to do this, we need to publish this one, because we have a lot of potential sources for by prospecting to produce products. And if we compare in South Asia, more than 13,000 species of plants are being used in traditional medicine and herbal cosmetics. And out of 13,000, which is 8,000 species, are natural products and contain reservoir of ethnomedical, ethnobotanic information. And for cancer, for example, this one in Vietnam, at least 25% out of 800 medicinal plant extract from Vietnam and Laos, for example, we talk Laos, so I don't want just to talk about Indonesia, just to be more balanced. And 13 were new anti-HIV agents, and 3 were anti-malaria agents. Yeah, so sorry, on the bottom, you see the plants found in the conservation area. I'm going to focus more on conservation area in Indonesia, which is the area under the Ministry of Environment and Forestry, because we have 55 national parks in Indonesia, plus nature reserve, plus other parks. Yeah, so agricultural area and others public areas. So the bottom of the slide, you see the nepenthes that we can extract or isolate something from nepenthes. Anyone know nepenthes? All right, knows the carnivora plants. And then on the right, on the bottom, it's actually mushroom grow in the elephant dung. Yeah, so this one interesting to explore more. So this one also, I mentioned already that Indonesia consists of mountainous areas, lowland, and freshwater marine. So there are still a lot of flora and fauna to explore, and also the microorganism. That is this one, for example. I'm putting here the social forest. So in addition to the conservation area, our Minister of Environment and Forestry has social forest and also the customary forest. So under this forest, they can also do bioprospecting things, as long as it's for the benefit of the community. Yeah, so I give you a list of examples that the community, the local community, using their traditional knowledge, they also trying to find products, for example, for natural coloring, for food, for cosmetics, and also the last one for drug or medicine. So I put the photos there. So actually, that's the Diamonorops draco. It's rattan jernang. Rattan is a kind of species of belongs to rattan. It's look like this, the fruits. Any of you tried this one for the foreign colleagues? It's under in the restaurant. It's actually, we call it snake fruit. Look like this, but this one is different, but the same, because of the family is the same Arecaceae. So those that you haven't tried salak, you need to try. So this one is very good for a lot of diseases, but still not scientific fight yet. I mean, not a lot of study to do that. Next. Yeah, that's so the Diamonorops draco is can be the indicator when we cannot find the Diamonorops draco or rattan jernang. That means the habitat already damaged. So this one example, I told you the tribes of Indonesia, more than 300 tribes. So I just want to link between this tribe and what kind of medicinal plant they have, and they already used from in the past. Yeah, for example, the top one from the eastern part of Indonesia, so East Nusa Tenggara province, that they have this faloak. So this one name of they call it something like ant, something, yeah. So, but this one also for antioxidant, antifungal, and so on. And then the second row is actually from Kalimantan. So that's people now heard about the bajakah. That's for anti-cancer. So this student actually won a competition in Singapore about the bajakah wood, but it needs to be studied further. And the bottom one is actually the tribe from Papua. We have all different traditional costume also, yeah. So this is not just for photographs, but they wear that one also. And it's famous for the sarang semut. So semut is ant, so like this ant, what you call cage, something like that. So it's Myrmecodia platyria, also for anti-cancers and others. Next. Yeah. So the another potency, this one in Lombok Island next to Bali. So we found that's in the Mount Rinjani National Park, a mushroom that actually only found in China, and then now in Indonesia, the morel mushroom, that's also potential for a lot of diseases. And then the down there is from North Sumatra, the Batak tribes. We have Taxus sumatrana there. So that's it's long history. The paclitaxel is alkaloid from this Taxus, but Taxus sumatrana is especially from Indonesia, while other Taxus in other part of the world. Yeah. Next.
Excuse me, Dr. Annie, you have 5 minutes left. Thank you.
So another in Gunung Ciremai National Park, Gunung means mount, yeah. So we found the PGPR, the plant growth promoting rhizobacteria. So this one can replace fertilizer or chemical fertilizer. So you use this one as a biofertilizer, and this one can also be anti-frost. So if we we plant potato plants close to the mountain, so to -7 degrees centigrade, so this the potato plants can be tolerant to that or resistant. And then it increased several commodities productivity, and then early maturation, increased growth rate, and reduce pesticide use, and increased tolerance to drought. So this one you can tell from the roots, and also the left one down is the control one during when it goes to -degrees centigrade. The right one is the resistant one. Next. Now, so this one from another conservation area, the Merapi National Park. They found also the antioxidant plants, and they already produce, but still small scale. And so next stage is actually for commercialization and maybe a larger scale and more scientific research. Yeah, so induced mutant, so this one can be through many methods. I just want to give you the research that I've done and still ongoing also at Rutgers last time, in here under Fulbright. So with Professor Ilya's team and also with Professor Jim Simon team. So we've done the metabolomic. And also I've done the genomic and also the transcriptomic things. That's in addition to Fulbright. So induced mutant can be from using gamma radiations and then light artificial color and chemical induced mutants. So on the right one, I just give you the figure of the targeted compound using the LCMS, and then the result is like that. It's actually red light influence the metabolite profile, while so it's affected by varieties, affected by the treatment, affected by the organ, so it's all affected by different treatment. It's very interesting result. Next. And next one, this one with Professor Jim Simon's team. So that's for capsaicinoid testing and also for capsaicinoid and polyphenol assay. So this one is very interesting that the control one, the non-treated one with colchicine, it's actually higher pungency, so you can taste that it's hotter when you eat the chili pepper. But when you treat it with colchicine, certain level of concentrations, it's less pungency. So maybe then you can eat more chili pepper without being sick or diarrhea or something, but you can get more nutrient from that. Next. Yeah, so this one, the transcriptomic things that study that I did while I was there, but it's not actually really the focus of my study that time, my research. So this one also, we tried for housekeeping genes and then 10 target genes. Next. And then this is the result. I don't want to just some of the results. There are a lot of results. So you can tell here for certain genes it's under down-regulated, so it's not expressed. But there are 3 other genes that are upregulated, so expressed in leaves and also in certain ripeness level of the fruit of chili pepper. And this one, the genotypic based sequencing I did also and still ongoing to be sequenced now. So this one is very robust, very high throughput one, and why we need very high quality of DNA also, and very complicated and tedious procedure to have that libraries constructed, and then later on we're going to send to the sequencing company. Next, so that we can fish mine the SNP depending on what trait you want to try and that, to study. So move on to the 4th one very quickly. Yeah, very quickly. Strategy and collaboration, and then this one actually, next. Yes, so scientification of jamu. Jamu is a traditional herbal medicine in Indonesia. So it's one a lot because a lot of tribes, but still a lot of things to do. And biopharmaceutical and phytopharmaceutical. So high demand of certain species. For example, the rhizome species. There are like 4 rhizome species that are on demand, high market. High demand, sorry, and then there are others that some of them are already exported by the government and the company. Yeah, so this one, how long it will take to produce drugs. It can be more than 13 years. And then for standardized phytopharmaceutical can be 7 years. For jamu, the traditional herb medicine, that also needs scientification study. So this one, we need to collaborate, that's why. Next. And 6 hexahelix collaboration, the government, the university, the industry, research institutions, NGOs, communities. So that need to collaborate more. And the components, the policy, funding, long-term program, funding also I mentioned earlier, that needs to be long-term also, and sufficient funding, infrastructure and institutions that need to be considered. And challenges, I just listed 5, but actually more than that. So how to change the mindset toward by prospecting, because they said all by exploration, as Professor Ilya mentioned. So they said, oh, it takes a long time, so the industry doesn't want to contribute for that, so they don't want to fund basic research. So this mindset needs to be changed also. And we need to set up collaboration platform, clear legislation, legal issues, regulatory framework, proper balance and benefit mechanism, and then future challenge is even more or even harder to address, but it's actually a combination of that 4. So way forward, our recommendations. So by prospecting, with the implementation of omics technology can be fastened using this technology. But a long-term program with sufficient funding is critical for explorations, research, and screening for drug development. Need to involve the pharmaceutical industries or company for funding also. And there's an issue of administrative, legal issue, and weak regulatory framework, especially in developing countries, need to be improved. And integrated approaches are required to boost the benefit from biodiversity for economic development. So with that, I would like to end my talk. So thank you very much. Oh, that acknowledgement, the last one, I need to acknowledge. So, so this one, a lot of, I would like to thank all people that already working with me and still been working with me. So thank you very much. Terima kasih is Indonesian language, and matur suksma is Balinese language. Thank you. Yeah.
Okay. Thank you, Professor Eni. Thank you for the nice presentation. I'm sure there is some questions from floor or from the online, but we will keep it for later. We will move to the second speakers. Please, Dr. Rongdi. But first, I would like to read about the CV of Dr. Rongdi. Please, okay, now I cannot read it. It's okay, I can read from here. Okay, Dr. Rongdi, she's an Associate Research Professor from the Department of Plant Biology in School of Environmental and Biological Sciences, Rutgers University, New Jersey, USA. Her educational background, so she obtained a BSc from Plant Protection and South China College of Tropical Crops, Hainan, China in 1983. And following with the MS in 1989 from Plant Virology, Iowa State University in Iowa, and also the PhD in 1992 in Plant Virology, Iowa State University, Iowa, USA. So honors and awards she obtained in 2005, 2007, 2009, 2015, 2017, 2020 from Rutgers Faculty Compensation Program Awards. And her research interest is plant biology and pathology, gene regulation, and molecular identification. So Dr. Rongdi, you have also about 30 minutes for presentation, so the floor is yours. Thank you.
Thank you very much. Thank you so much for the invitation, especially to Professor Eni and Dr. Ilya Raskin. Really, sincerely thank you for inviting me to this fantastic talk, this fantastic conference, and I'm just very grateful to be here. And growing up in southeast part of China, I have known many of my neighbors with relatives in Indonesia, Malay, Singapore. My husband has relatives in Philippines, but I have never been to any of this country. So it's great to be here. When I came here 3 days ago, I saw banyan tree with the aerial roots coming down from the branches. I said, I'm home, because that's my name. Rong means banyan tree, and I was born in a city called Fuzhou, the capital city of Fujian province. So, and then I saw banana and coconut trees, and I said, that's where I study, on Hainan Island in the South China Sea. It's tropical. So I'm feeling like I'm home. So thank you so much again. At the request of Annie, she asked me to present 2 parts of my research related to gene editing of plants or genetic engineering of plants for disease resistance and stress tolerance. And the second part, I will talk to you about our work on using C. elegans, a little tiny worm, to study health-promoting compounds from plants. So if I may have the next slide, please. So first of all, gene editing for plants to improve disease resistance and stress tolerance. So next one. We all know that human depends on plants, and we depend on them for food, fiber, medicine. Animal depends on them for feeds. So next time, and it's very important to improve the plants. And as we all know, it's a human started, that the human actually is very much involved in the crop improvements. And I give you an example here. This is a wild corn, and to have the modern corn, it takes years and years and years. So it's too long. It takes too long. As the population grows in the world, if I may have the next slide, please, that we need to have better ways, faster ways, more efficient ways, more specific ways to improve our plants. And as we all know, to do the traditional breeding, we are very limited to the gene pool from our plants. And so that actually gives us limited breeding options. So here comes the biotechnology. And we normally use the term GM, genetically modified plants. I often say we are all genetically modified organisms, including ourselves. So with genetic modification, genetic engineering of plants, we can actually move different genes from different organisms, from even animals. Animal genes can be put into plants. So it's much faster, and we can be quite specific. Of course, the issue of GMO or genetically modified organisms is not unique in one country. It's a global issue. Okay, so next slide, please. So it's a plant pathologist that I was always looking for ways to improve plants for the disease resistance. And I started with making transgenic plants expressing co-protein genes from a virus, so we can induce the disease resistance. Of course, that comes the GMO issue. So, and I started looking for other ways, and I'm like, any other ways that I can use to make plants resistant without the transgene issue. I started looking at zinc finger, then I was looking at TALEN, and then I was ready to do TALEN technology. Here came the CRISPR, and I was telling my students, let's forget about TALEN, we need to start with CRISPR. So I'll give you a very, very brief overview what CRISPR gene editing is. So it needs like 2 little components, one small one, one big one. The small one is called the guide RNA. As you can see from this diagram, the top is, this is a diagram for CRISPR gene editing in animal cells. So we can express the guide RNA in a separate plasmid. It's very easy. You can make them in the test tubes too. And then you need the Cas9. Cas9 is a big enzyme, it's a big DNA. You can express them in a plasmid. Or now there are lots of them being purified. So you just need the enzyme and the guide RNA. You introduce that to the cells. The bottom diagram shows you how the guide RNA binds to your target site. So as we know, the longer of your target, the guide RNA, the more specific it can be. The shortest you can make is about like 19 or 20 nucleotide. So, and that it's connected, linked to, if you see the blue scaffold, it's called the scaffold. It's 80 nucleotides. In any organism, you can use this: bacteria, fungi, plant, animal, anyone. That's what Jennifer Downer and her group and her partner Charpentier, they figured out how the Cas9, this endonuclease DNAs, bind to the scaffold and being dragged by the guide RNA to the target genomic DNA. Because the Cas9 need to hop onto this scaffold. You see the blue, two big blue, and those are the representative of the Cas9, this enzyme. It will come to the specific target sequence. And then you see the two scissors there, and I cut at a PAM site. It's any nucleotide plus 2 Gs, NGG. So it's quite common that you can find NGG almost anywhere in a small stretch of a DNA. So then the Cas9 will cut at the specific site where you design your guide RNA. It's as simple as that. So because of their work, they won Nobel Prize in 2020. So next slide, please. And once the guide RNA, the Cas9 comes to the genomic target site, the mutation occurs, because when DNA is damaged, any organism will try the best to repair the DNA. So the repair is actually, most of the time, it's perfect, because the cells will have to survive. However, during the process, there are two mechanisms that the cells will try to repair. One is by the non-homologous end joining, the other is by recombinant, recombination, homologous recombination. So if there's any DNA nearby with the homology to the target sequence, that DNA can be flipped or replaced into the cut site. But non-homologous end joining also, mistakes happen. Often you lose one nucleotide, you lose two, you lose a couple of them, or you have insertion. So it's called indel, insertion or deletion. Then you have a gene disruption or mutation. Because of this, CRISPR is made to be very, very specific and kind of an efficient way to introduce mutation. So next slide, please. So why I was so excited about CRISPR is because I saw the opportunity for us to engineer the plants and make a mutation in the plant genome itself. And the possibility is also that when we engineer plants by transgene approach, you introduce a gene to induce resistance. That resistance can be overcome by the evolving pathogen. If you engineer your plants, that resistance stays for much longer and stronger. That's one. And two, the process of doing CRISPR gene editing, there's an opportunity for us to, one, segregate out the transgene, because the transgene here is the guide RNA and the Cas9. The mutation site and the transgene integrating site is two different sites. And so you have the opportunity to segregate out by the following generations of the plants. Or you can use transient DNA that does not integrate into the genome. Or the third method, I told you, all you need is the guide RNA and the enzyme. RNA and enzyme, once you introduce into the plant cells, they are not very stable. After it's done its job, after I heard the talk from Jennifer Doudna, it takes about 4 hours. Actually, it's short, it's about 15 minutes. The gene can be damaged. So then when the enzyme and the enzyme and the guide RNA got degraded, they disappear from the cells. But the mutation already occurred. So I see this as an opportunity to engineer plants without using the transgene. Next slide, please. So I'll give you an example. I'll talk to you about Fusarium haploid. This is a disease that I have worked for many years, and the toxin that they produce. It's one of the examples is deoxynivalenol, or we call DON, and it's the fungus. It's called Fusarium graminearum. They infect wheat and barley. Wheat is like number 3, very important plant in the world, or number 2 if you think about it. And barley is the one that we use for animal feeds and use it for making beer. And so any wheat and barley in the whole world, there's no resistance to Fusarium. There's only one kind of wheat from China that has some resistance to Fusarium haploid. So as I studied, we understand how this toxin, DON, affects the cells. It interacts with the peptidyl-transferase center of the cells that's involved in translation of the cells. If your translation is interrupted, your cells will die. So it causes apoptosis. So it interacts with this protein called ribosomal protein L3 in the ribosome. Next slide, please. And if I may have the next slide. So we understand how to interact with the ribosome protein L3. What I did was initially, I took the yeast RPL3 gene, only parts of it, and engineered that into wheat cultivar. And we made lots of transgenic wheat, and I can germinate them on petri dish on the left. You can see the seed got germinated in the petri dish containing the toxin. The toxin is a pathogenesis factor. So if the plant can resist the toxin, they will germinate. So you can see on the two on the right, it's Rutgers. We named it Rutgers cultivar, and they're transgenic, but the one on the left is the wild type. 7 days after, they will all germinate, but you can see the size difference. 10 days after, the wild type will die. The transgenic will not. So we have great resistance. We test them in the greenhouse, we test them in the field. You can see we can inoculate. And this fungus, when they infect wheat and barley, and also corn, they infect the immature inflorescence, the florets, the immature flowers. So that's why when they infect, and the fungus and the toxin stay with the grain, and it actually causes a lot of trouble, as shown in the previous slides, and it makes people sick, and also livestock, they get very sick. So now you can see our transgenic ones, they work great. It only where we inoculate by injection of the spores into the flower heads that got a little bit brown, but the wild type will totally die off. So we have patents, we have publication, we have companies from DuPont, from Novartis. I remember they came to us, wanted to get our patent, and then after a while, they stopped contacting us. It's because of the concern of GMO. So it's because of this work, and I was like, we spend so much time, so much money making great resistant plant, but it's impossible to be applied and given to the farmer to grow because we're still concerned about the GMO. So next slide, please. Then I told you about the CRISPR. So once we decided to do this, and since I'm going to manipulate cause mutation in the plants, I specifically want to look for something we call the disease susceptibility genes. So those susceptibility genes are host factors. They interact with the pathogen, whether it's fungi or bacteria or virus or even nematodes. If you knock that out, that would not, if it won't result in the penalty on the plant growth, and that can knock out the interaction between the plant and the pathogen, and that can stop the infection or prevent further infection. So one of those factors, I call it the 2OGO gene. We use the Arabidopsis as our model system to test this out. And because I have seen EMS induced mutants of Arabidopsis, and then they had those mutants, and they were screening for downy mildew disease resistance. At the same time, they checked the Fusarium, and there's one little report saying that they do resist Fusarium infection. So I said, okay, we take this as the first gene because it can induce the salicylic acid pathway. It's all plant pathologists know that this is the pathway that involved in the upregulation of a lot of the plant immune factors, and that can resist plant pathogen. So we made our own vector. If I may have the next slide so I can go faster. So we made the guide RNA and the Arabidopsis U6 promoter and the Cas9 and the Arabidopsis ubiquitin promoter. And of course, this is an integrating vector. And then we choose the target site after scanning through the genome, and you can see the CGG at the PAM site. We have a restriction enzyme site right before it. It starts with the G because it's a U6 promoter. And we choose the restriction site there because later we can use the RFLP, which I will show you really quickly, to screen our mutants. So next slide, please. And so here's one example of the RFLP. When the restrictions at the target site is mutated, I lost the BAGO2 restriction site. If the wild type would do PCR, amplify out the target region of the genomic DNA, and so it's also from the mutant plant. So if the BAGO2 is destroyed, when I want to digest the PCR product, it will be digested into two bands. You can see on the right, the wild type, and on the mutants, however, they should produce 3 bands, which is very common because most of the time it results in monoallelic mutation, meaning one allele of the gene is mutated, the other allele stays as wild type until you segregate them out in the next generation. So when the allele is mutated, then you get the top band, which is the whole sequence, or maybe minus 1 or 2 nucleotide. And we use another assay to check it too, so the gel pattern is a bit reversed with the wild type, which are the two common methods to check for mutations initially. So next. And then we screen for the next generation, and you can see we got homozygous mutants. Next slide, please. And you can sequence them. You can clone the PCR products out from the T0, T1, and T2 generation. So what we want to look for, and you can see the target sequence in the middle. You can see the deletions, the insertions. So we try to look for those that totally disrupt the open reading frame and result to even stop early stop codon of the gene. And then we, at the T2, we select for the ones that we have lost the transgene, because you can easily use a PCR to check them out. And then we have the good mutation. So next slide. I think this one has a little bit more, just keep going, more. And so we've chosen that one, and go to the next slide. And so what we wanted to do is then we test for the resistance. I will show you the slides together. And since I studied Bali, I already made transgenic wheat that's very resistant, but now I wanted to work on Bali. And what I want to know if the equivalent gene of Bali works as a susceptibility gene or not to Fusarium haploid. So I cloned the Bali gene, and then I put that back into the mutant in Arabidopsis and see if the disease susceptibility came back. So next slide. As you can see from the diagram, that this is how we grow the fungus, and we inoculate the little florets from the Arabidopsis on agar plate, and then we inoculate them. Next slide. Then you can see the top row shows you the wild type of Arabidopsis florets. After day 2 or day 3, they became sick already. By the time it's day 6, they're totally destroyed by this fungus. And the mutant, however, stay pretty nicely until even 6th day. And the Bali 2 gene complemented a mutant, and it regained the susceptibility to Fusarium. That tells you this gene works just the same as the Arabidopsis ones. So it works really well. And next slide. And of course, we check for the disease infection. And so you can see the mutant had pretty good resistance. So next slide. Because this is published, so you guys can check out the paper. So I'll go really fast, and we check out the salicylic-related genes, and you can see those genes are upregulated. That's what provides the disease resistance. So next slide. Next. And so with that great success, we moved on to the next disease. In collaboration with the great Dr. Jim Simon here, I learned about basil. And in China, I never had basil. Southern part of China, only really south, Canton maybe a little bit. They eat a little bit basil, but I never even smelled basil. And it was this wonderful crop there. And I learned about the downy mildew disease. It's caused by the fungus called Peronospora balbrii. And it's a nasty disease, actually. You can see on the graph on the photo on the right, the crop can be totally destroyed. And so we know about this fungus. Next slide. And so we choose the gene. Actually, 2OGO gene is also chosen. And this one I'm showing you here is the other gene called homoserine kinase, HSK. One of the mutants from the EMS library also show it has great resistance to downy mildew. So we scanned the transcriptome and the genomic data of the sweet basil of our breeding line. We call it SB22. And we clone the genomic DNA, and we choose the target site, and we build our vector. We use a gene gun, and this time I deliberately made the transient vector because this is basil. We want it to be, in the future, we can commercialize it. So I don't want any transgene if I can help it not to be in it. So we use a gene gun. We can also use agrobacterium to put our integrating vector into it. So next slide, please.
Excuse me, Dr. Rongdi, you have 10 minutes left. Thank you.
Thank you. Okay, 10 is better. Otherwise, I thought I had like 6 minutes. So Then you want to engineer any plants, you want to have a good tissue culture protocol. If you cannot regenerate plants, yes, you can test all of your genes, but you can never regenerate a plant out, you cannot have the final products. So with basil, I thought it's an annual crop, it should be easy. It turned out to be not so easy. It took us quite a while, and we finally found an explant that we can work with by manipulating the different hormone combination. We successfully transformed it, and we can regenerate it. Remember, that's a transient vector. I don't have any selectable marker, so we screened hundreds and hundreds of them, and then we finally got the plants. Next slide, please. So we did the same thing, RFLP, sequencing, and we have many mutants. Next. And we test the mutants out in the greenhouse. And so you can see, compared to the wild type, they were quite resistant. Next slide, and we published this. And then we test them on our farm in the natural environment. And as you can see here on the top panel, a month after, you could already see the difference between the SB22 cultivar and our mutant line, and Dolly, the most susceptible line. And by 2 months later, look to the right, Dolly is like completely gone, or not even one piece of leaf left. And wild type SB22 was also quite sick. Some of our lines, not only the disease onset was delayed by more than a week, but also some of them were still kind of standing there at the end of the season. So we dug them up. Next slide, please. We put them into the greenhouse and we collect seeds. We're going to do the second year testing. So I want to summarize this a little bit on CRISPR gene editing of modification in plants. The gene silencing is targeted and precise. The gene knockout is more efficient, a lot more efficient than RNAi technology. It's a total knockout in most cases. And the host factor can be knocked out to prevent pathogen interaction with the plants. Hence, we get disease resistance. Okay, and potentially we may not need to worry too much about the GMO because I don't have any of the foreign genes in the plants that we generated. Okay, so next slide, please. Now I want to give you in the next 8 minutes or so about Sialogans. What do we use this for? To study the health promoting compounds from plants, natural products, and what I would like to combine these two sides of our research together to engineer plants. Okay, so next slide, please. What is Sialogans? Lots of you may know already. It's a tiny little worms. It's about 1 millimeter long, transparent. What's good about it, it's one of the first multicellular animal that's gotten the whole genome completely sequenced. We knew almost every single cell of this little animal. What does it do? We know we know about almost all of the genes, each one of its function. And the most impressive about this little animal is in 2.5 days, it can go from egg and become L1, L2, L3, L4, and adult and lay eggs again. It takes only 2.5 days. So it gives us enough time to screen what we want to screen and study the genes we want to study. And also in a very short time to go through the next generation. And most important thing about the Sialogans with human health is it has lots of the genes that's similar to human genes function. And I give you an example here. The insulin signaling pathway is almost the same as that in the human, compared to 1 millimeter long little worms. So we take advantage of this, and we can use that to study any compounds, any chemicals and medicines, and to look at the effect for human health improvement. So next slide. So I use that to study diabetes and Alzheimer. I'm gonna go very fast, and this is a problem in many, many of the countries in the world now, not just developed country, but also developing country and also China. So next slide, please. And I will go, and this type 2 diabetes, and next slide, I give you about the medication on Alzheimer's and Alzheimer's disease, and diabetes are quite related. So next slide, please. So I can go faster. So the hallmark of Alzheimer is the beta amyloid peptide that deposited in the neuron cells, and that inhibit the neuron signal transmission. So next slide. And next slide. And medication for Alzheimer's disease is memantine. Okay? And we know specifically its function, how that works. But mostly it's to alleviate the symptom just a little bit. It's really not a cure. Next slide. And for diabetes, commonly used is metformin. The function of it mostly it's antioxidants. I said, well, it's antioxidant. We have plenty of plants. We have plenty of compounds from plants, from our food that have antioxidant activity, right? So next slide. And so I started using shea ligands. I can buy transgenic shea ligands that express amyloid beta with $7 a strain, and that was sent to me. This one specifically is the amyloid is expressed under the temperature-sensitive promoter, so you can only grow the worms at 16 degrees, a little bit cold, and then if you bring that to room temperature, we set to 22 Celsius, the amyloid beta will be expressed. They will become paralyzed. In about a day and a half, they will die. So next slide, I will show you. So you can see that to the top panel, the shea ligands are grown at 16 degrees, and they make a weird movement. Normally, the wild type on the left, they make an S-shaped movement. The shea ligands, the transgenic ones, they make a circular movement. But once they die, they become a little line, so you can easily score them. So next slide, please. And within 36 hours, they will die. So in collaboration with my friend, Dr. Chunling Long from originally the Kunming Botanical Institute in China, and we look at Lycoris plant. It's a beautiful plant. It has flowers in the spring, and the flowers gone, then you'll have the leaves. From the bulbs, it has a lot of bulbs, we can make extraction of all sorts of alkaloids. So next slide. Some of those compounds, some of the alkaloids, they inhibit, they are called acetylcholine esterase inhibitors. And the choline balance in between your neuron cells is the most important thing to maintain the neuron health. You cannot have too much of it, you cannot have too little of it, right? So galantamine is one of those type of alkaloids, and that's not used in the US, but it's used in the European Union to treat Alzheimer's. So I was like, why can't we look for more of this from the natural compounds? So next slide. And so not only we use the natural ones, we also made the synthetic ones. Next slide. And so why to study that? I already told you about it. Next slide, please. And so we treat the worms, and we compare the compound. If I can elongate the lifespan of the transgenic worm after I took them to the room temperature, if I can extend the life, then that means our compounds have an effect in inhibiting amyloid beta expression and the deposition, right? So we did that. So next slide. And we found out, indeed, those compounds that we isolated from Lycoris bulbs, and that can have that kind of function. And then we studied whether it really inhibits the acetylcholine esterase, the gene expression of it, and then also later the protein level, and we could see that from our compounds. So next slide. And not only that, the Lycoris, those compounds are involved in suppressing the stress-associated genes, inflammation-related genes in shea ligands, and those are the factors that contribute to the lifespan extension in the shea ligands. So next slide. So with this, what I want to tell you is, next slide please, that using shea ligands, it's a very convenient, simple method that we can study, we can screen all of the compounds. For not only anti-Alzheimer, we are also looking at anti-diabetes effect, anti-obesity. We can also study wound healing. So for any compounds that it has own wound healing effect or anti-UV that can be incorporated into cosmetics for lotions or sunscreening or wound healing for wound. So with that, we have patents, we have publications. It's a very useful little tiny animal model system. Otherwise, the next step, you go to use the mouse, and that's quite expensive. With the C. elegans, it's very easy to study the molecular mechanism of any of the health promoting effect from your compounds. So next slide, please. And I give you a little bit more on the other project that we embarked on, and we already published that too, on vitamin B12 using C. elegans. Vitamin B12 is very important, and you get that only from animal products, or you take the supplement. So, and it's highly associated with Alzheimer's. So once I learned that, what we made was, next slide, we made C. elegans that's vitamin B12 deficient. It was not easy, but we made it. So next slide. And I want to apply to a compound from the Chinese there. It means luo han guo. It's a Buddha fruit from this fruit called Momodica grosvenori. It's grown only in the Guilin Mountains in China, in Guangxi province, and they make tea into it. For us, we normally drink the fruit. You put it into hot water and take out the glycosides. So next slide. And the glycosides, there are 5 of them. It's called mogrosides, 1 to 5. 5 it's the one that's most effective. And it has 0 calorie. It's 300 times sweeter than sugar. So it's being used as a sugar substitute. So next slide. It has not only doesn't have any calorie, it has great anti-inflammation, anti-cancer effects in cells, and also I did that in cells and also in animal model. So we know that. I want to see if it can help with the vitamin B12 efficiency. So next slide, please. And so I applied that to C. elegans, and I know it can extend the lifespan of vitamin deficient C. elegans. So next, under different conditions. And also it has great antioxidant activity. With the C. elegans, it's very easy for us to collect them and measure them. So next.
Excuse me, Dr. Rondi, your time actually is up.
About 2 minutes, if I may. So we studied the gene expression related to oxidation, and of course it has great effect. So next, and a couple of slides. And we have the amyloid beta and transgenic. The amyloid beta is expressed in the neuron cells, and this assay takes only 5 minutes. But of course, you had to count at least 30 worms of them. So our paper has been published. You guys can check that out. So next, I think that's the end. Okay, so with all this, I think our lab with several students and postdocs involved, and my collaborator, Dr. Michael Lawton, and we had grants from for the plant part and the animal part from USDA, USW-BSI, and our agriculture experiment station. So, and thank you very much for your attention, and I suppose that we can take questions later. Thanks a lot.
Okay. Thank you, Dr. Rondi. Very interesting topics. But we still have 3 speakers. So I will move for the third one is Dr. Ines Slametludin from IRI, Indonesian Rice Research Institute. Dr. Ines will bring the topic of tapping diversity for crop improvement through gene editing and transgenesis application. But before that, I will read about Dr. Ines CV. So she's currently is the global leader of CGIR Gene Editing Initiative and head of Genetic Design and Validation Unit at IRI in the Philippines. She has been working on rice genome engineering for more than 25 years. Prior is the head of Molecular Biology Division at LIPI, or now is BRIN. Educational background of her is she got postdoctoral fellow at the Institute of Molecular Plant Science in Leiden University. She obtained PhD from Nottingham University, UK, and got the BSc from Agriculture IPB University in Indonesia. Some honors and awards, awarded as the Best Young Scientist of Indonesian Institute of Sciences at LIPI, a medal from Indonesian Ministry of Agriculture, award from Indonesia President for Food Security Contribution through Biotechnology, the most cited scientist at RC Biotechnology at LIPI, And at IRI, she won the Best Scientific Achievement Team in 2017, and also received the Best Scientific Team Award from the DOST Los Banos in 2018. Her research interest is the nutrition improvement, disease-resistant drought, low phosphorescence, and yield improvement in rice or golden rice. So, Dr. Ines, same, you have about 30 minutes for presentation. Thank you.
Thank you very much for the nice introduction. Good morning, everyone. And I wish to thank Professor Master and also Dr. Saptowo and Dr. Annie, Professor Annie, for inviting me here. It is always great pleasure to be back here, and particularly in Bali. I was tempted to get one flower there to put it here to give some of a Balinese spirit. I used to be a Balinese dancer teacher in the good old time, many years ago. And they said if you, when you grow older, you involve in art, you make life better. But I decided not to do that because I'm afraid that Dr. Tini would get mad if I ruin the flower there. So let's, my talk today is about typing diversity for crop improvement through transgenesis and gene editing application in rice. Next, please. Initially, I was gonna drop the transgenesis topic, but I was inspired by Professor Simon yesterday talking about nutrition. If I missed the transgenesis part, there would be no nutrition aspect there. So I'll talk very fast on the transgenic part. If it's too fast, please blame him. So the talk would cover three aspects. It is transgenic approach utilizing traits from other germplasm genes, transgenesis for gene function validation very fast, and gene editing to develop non-GM products. Next, please. Okay. Now about the transgenesis. Next, please. So if you look at the globally, the anemia prevalence and zinc deficiency is actually major, bigger problem. 1.72 billion, around 50% of it is anemic, and around 50% of it is about due to iron deficiency anemia. And for zinc, 160 million people are stunted to zinc deficiency. Stunting is not only because of zinc deficiency, but it contributes quite a lot. So the potential problem solving is diverse diet, supplementation, fortification, and biofortification. And from the reality on the history, we know that no single approach was successful. So next, please. So the principle is like not to encourage single-based diet. Many people said, oh, why talk only about rice? But the philosophy is why we eat an empty rice. So if you can make the base diet to be richer in nutrition, it would be better. Next, please. So the product concept of high iron and zinc rice is we reach iron concentration more than 10 ppm, which is 5 times the baseline, which is 2 ppm. Zinc concentration more than 30 ppm, which is current baseline is 16 ppm. And the yield should be within the range of the conventional counterpart. And we will stack it with the golden rice through market-assisted breeding. We have to have a good grain quality and also stack with farmer traits. And with the first target country is Bangladesh, Philippines, and Indonesia, and we are potentially taking it to Nigeria and Ghana. Next, please. It's a long time to develop the transgenic because you start with the proof of concept. We publish it on the when we attain the first iron and zinc. And initially, we combine the rice gene with the soybean genes, and because we were at that time was advised maybe it's easier to not to use soybean because soybean is the source of allergen, although it is not the gene that we're talking about. But so we changed it to apple. So 1 apple a day makes the doctor away. I think, so we use the gene from apple, but we use two different promoter. The first promoter is expressing on the grain, which is the outer part of the police rice, and the second promoter goes into the dipper. So we use the two gene for the ferritin, which is, and then one gene for nicotinamide synthase. Nicotinamide synthase basically is a iron and zinc chelator. And now, all the way through, we started in 2010, and now we have the advanced development. We have multi-location trial in Bangladesh. We have a multi-location trial in the Philippines, recently finished. That was for the regulatory aspect. Next, please. So when I was in Indonesia, before I used to work with the BT rice, and I was thinking that if I developed 10 events would be good enough. But in reality, we developed more than 1,000 events to eventually select only 2 events, which is with all clean this, a single copy and everything that would make it good for the regulatory process. So in 2001, we did an 8-location trial in Bangladesh. In 2020, not 2001, 2021. In 2022, we did a 5-location trials. And then we also just completed 2 location trials in the Philippines. Next, please. And the yields about more or less the same, about 6 tons per hectare during Boro season in 2022. Next, please. And then we were so happy when the iron and zinc concentration is really, really good, that it was more than 30 ppm for zinc, which the current breeding efforts for high zinc reach about 24 to 25 ppm. And then the iron, we reach in the Philippines is around 10 ppm, but in Bangladesh, because the varieties cycle is longer, on the Boro season is 155 days, so we got more accumulation from that. Next, please. So our colleague in Harvest Plus, who is a nutritionist, did a modeling. Basically, the message here, if you have the prevalence about 100%, and then you replace your rice with the high iron and zinc rice, you can reduce the prevalence from 199% to from 6.6 to 12%, depends how much your caloric intake based on rice. So, and this is actually more than fortification of 35 ppm, which is the current practice now. But in some cases, there's a fortification of 50 ppm, then it's less. And you compare with the zinc breeding, which is the right side of the histogram, that was the reduction of prevalence. So zinc breeding is good enough, but it reduced the prevalence by 20% around. But if you replace it with the transgenic high iron and zinc, you can reduce the prevalence by about 70%. Next, please. Next, please. Okay, so we have a collaboration with Professor Ella Tako from Cornell University. Next, please. On a feeding trial on chicken. I mean, a lot of people ask me, why chicken, why not mouse? But the chicken, basically, I don't have much time, but it is a good model because of this broiler model mimics the physiological effects of FA deficiency, as reported, and it's a low cost. Next, please. I think I just snapshot one of the interesting results that we didn't expect is that you actually got a good bacteria population here. If you compare, this one is the 2 transgenic on top from Philippine rice. We give actually the chicken Philippines diet, and then the chicken is also another subset of chicken eat the Bangladesh diet, which is maybe they're happier, sorry. And then you can see the transgenic one, which is this one, is you have a lot more population from the useful bacteria, microgut bacteria. And you know, the microgut, on the other hand, increase the bioavailability. So it's a very good cycle, and as we know, a lot of other good things there. Next, please. And the idea is to be combine that vitamin A, iron, and zinc deficiency in biotech rice. So we will combine it with the golden rice. Golden rice is currently has been commercially released in the Philippines, but it at the stage of now that the farmer association is increasing the seed for deployment. So if we can get this 3-in-1, it would be at least to replace, for example, of course we could never expect that they would replace everything, but it would be significant impact would be obtained with no extra cost to the consumers. Next, please. So in summary, GR2 even golden rice has full regulatory authority for in the Philippines, yields is the same, and it would the same iron and zinc rice meet goals of 30 to 50% of estimated average requirement of women and children. And we are breeding it to the newer popular varieties, and with the farmer management exactly the same with the existing variety, and farmer and consumer study have been conducted as well to guide development. And on the Haizing rice has been undergone field trial in the Philippines and Bangladesh, and we aim to have a dossier completion by this year. Next, please. Okay, I just want to give you one slide, but I think it's unique. But when we did a transgenesis for gene function validation, which is eventually the product would be developed by marker. So you just do it to get a gene-based marker. You knock out the gene, you overexpress the gene, and then you get a very good publication. You can publish even at that time in Nature, PNAS. Our team working with the breeding teams. But if you work on the product, it takes so long, and then you send to a big journal, they will say this is not novel anymore. You can still publish in the good journal like Scientific Reports, but no way you can publish in this, which is a bit of irony, because the other one has probably more impacts. But next, please. So I think you can read that one. But now I want to talk about the gene editing. Most of our product of the gene editing aim for SDN1 and SDN2, which actually we also do SDN3 for the Philippines. Because in the Philippines and Kenya, for example, if no transgene, non-GM. But most country, not most, but many other countries, the decision tree, if you do SDN1, SDN2 is the one that is considered as non-GM. To show you that this, for those who are not familiar on this place, on this technology, there are already gene editing product in the market. For example, the Japanese aim for the consumer trade, the high amounts of GABA, so you feel good when you eat tomato, because it works with nervous system. And for corn, for example, increased starch content of, and then pairwise soon will lead to commercialization of green leafy vegetables, and rapeseed, soybean, improve oil quality. So there has been, although gene editing in the market, but of course it's mainly in US. Next, please. Okay, about regulatory environment, I think Prof. Pras already mentioned about it, which is great, so I just want to touch base on it, that if we look, this map used to be red, and now, even last month, I think UK already decided that most type of gene editing is conventional breeding products, so the map is become greener, that showing that gene editing would be regarded as a conventional breeding products. And even in EU, what I heard, at least what I heard, like I hope it's that in June there will be a meeting to visit the directive, because the most ministers of agriculture prefer that the gene editing is not regarded of GMs. I mean, everything is long in EU, but who knows? It is June there would be something happen. Next, please. So, in my recent task as a global lead of gene editing projects initiative in CGIR, I just want to have a touch base a bit. CGIR is working on rice, but CGIR are a consortium of a different institute working on agriculture, so including CIMMYT, including others, and in CGIR we work with banana, cassava, rice, maize, wheat, potato, although the current ongoing projects is also include the crops from dryland agriculture, which, including like pearl millet and sorghum, others. Next, please. If we talk to the breeders, most, actually last week I was listening to the professor from Minnesota. I think he's one of the best maize breeding. But the breeder tend to go to, in rice, tend to go to elite to avoid linkage tract. They exhaust the core collection of the breeding. So it's rarely now like the land races goes into the breeding, or even the wild rice species. Because it takes time. It's basically because you get a good one and allele combination, and it's easier. And taking out the non-desirable allele would take many years. So gene editing can avoid that and bring actually more diversity into the breeding scheme. Next, please. Rice, I think I accidentally deleted one rice. If we look of the rice biodiversity, IRI with the partners in China and other places has sequenced 3,000 genome of rice. And we have like one SNP every 18 base in rice. So there was so many diversity in rice. At the same time, it's also showing that mutation happen all the time. So when I just work for IRI, for example, the most popular variety globally, IR64. You grow next to each other, IR64 from the Philippines, and then IR64 from other countries, you can immediately see there has been already difference. They both call IR64, but the difference in sequence is even on the phenotype. So, okay, so I think Professor Rongdi already mentioned a bit in the diagram of gene editing in rice. Basically, we have to know, of course, the target gene, target trait, design single guide RNA, create DNA factor, and do reagent transfer either via agrobacterium, via a virus co-infection, a bombardment, electrophoresis, and we do need to have a system for tissue culture in planta transfer or tissue culture system. It could be changed in the future, but then when we do mutation screening that was mentioned about T7 assay or surveyor assay, and we sequence the target to make sure that we really got the mutation that we want to, and then conduct self-pollination or backcross, select population with the variant mutation, but the absence of the Caesar, because both were introduced into different locus. And eventually got the no transgene lines and do phenotyping of it. Or you can, of course, do phenotyping earlier than that. Next, please. Okay, I just want to show you some example of the genome editing work that we work with different partners. And one of them is this BLB, or bacterial leaf blight, so it's called CRESEC, which is the major disease in Asia, South Africa, and Africa. And it's a major constraint for rice production. And on the worst day, scenario can causes 20 to 70% yield losses. And these bacteria basically release their TAL effectors, where even these TAL effectors bound to the promoter of the rice sweet genes. There are 20, I think it's about 23 sweet genes. And then they bound to promoter of a few sweet genes and then eventually told the rice to produce more sucrose, and the rice dead, but the bacteria is happy. Next, please. What we're trying to do, the breeders actually have seen as well, and I think there is also work here, that there are using marker technology. You have, like, for example, the XA21, the XA1, XA2, which separately you have mutation already present in the diversity. However, it is challenging to combine multiple mutation because bacteria quickly, not quickly, but within 1 or 2 years, then overcome one mutation. So the idea with the gene editing, if you can do multiple mutation in one go. So we conducted a mutation on the sweet 11, sweet 13, sweet 14. The sweet 14, there are more than one tau effector. So 4 mutation in the promoter of the rice sweet genes. Next, please. And we got a resistant, broad spectrum resistant, and it's transgene free on the IR64 and the popular variety of Indonesia, Ciherang Sapuan. And this has been published too, and we continued working with different background of popular varieties. And also, I mean, you need to also think about different strategy, because with the CRISPR-Cas, we tend to get like 1 or 2 SNPs. But it is great for the regulation, but then there is also possibility it would be the disease could mutate faster. So we also develop lines which has a longer mutation. And for these, CPF1 tend to do a better work. So yes, and then so this work, that lines actually has been declared as non-GMO in US and also in Colombia, the BLB rice. So, and then the other work that we had done at the early stage of this technology is that Tungro virus. I mean, Tungro disease is one of important disease as well, and it in Makassar, di Ujung Pandang in Indonesia, because in the past there was a high incidence of Tungro, so there was a mutation in this cultivar called Utrimera, so which is resistant to the 2 type of the virus vector of the Tungro disease, the RTSV and the RTBV. So we need 7 backcross to cross it to Korean cultivar. If you think that's 1 backcross 2 times per year, then you need 3.5 years. So with genome editing, we obtain resistance to RTSV within 1 year. Next, please. But because scientists already know that the resistance to RTSV is controlled by EIF4G in the chromosome 7 of rice. And so what we develop is variant. So we do now have a multiple allele, which is resistant to RTSV. But I have to also share, because in some cases, if you knock out the gene totally, if we knock out this gene totally, rice needed to stay. So it's actually lethal. But if you just knock down the expression, then the infection is not happen, and you got the resistance without affecting the rice plants. Next, please. So, and currently we are working for the blast disease. This is actually mimicking what has a success in barley. Just changing 1 nucleotide of 1 gene, it is an LR67 in barley, it's actually confer resistant to multi-diseases. And what we're trying, we first now developed to test the effectivity in rice. We did a transgenic approach. We developed a vector with these mutated genes and introduce it to rice, and we did got a very interesting result. And now we are doing the gene editing approach. However, what success we got the transgenesis, of course you still have the endogenous gene. In addition, we got the mutated genes, right? You got two. So we don't know if we just edit it without the endogenous genes present, we still got this resistant blast. And blast disease is a big problem now. It used to be only in the upland rice, but it's also moved to also lowland. And in the future, if we want to grow more direct seeded rice, which I think inevitable because of, I don't know, those who are not familiar with rice, to grow 1 kg, to get 1 kg of milled rice, you actually need 2,500 liter of water. So in future, most likely it would move to the direct seeded rice, which, or one need to regulate when the water is needed for rice, because you don't need to flood it all the time.
Excuse me, Dr. Ines, you have 5 minutes left.
How many more minutes?
5 minutes left.
Good. Okay, so next, please. So then we also work with the other two traits is high nitrogen use efficiency and low methane emission. Both is for climate change mitigation. I won't go first. And we identified the gene and cadentate allele, and we also have checked the mutation is actually reduce the need potentially of fertilizer by 30 to 50%. But of course, this is a pot experiment. You need to check it in the field. It's not always the same case. Next, please. So, and also we work on the yield trait, the TGW6. This is increased the 100 grain weight. And we also work on the GN1. GN1 is increase the number of grain. But all these things, there is always pit and fall too. Because with GN1, turn out that most breeders is already selecting a modern variety, the knockout version. But if you work with the local varieties, then it did increase because it was not selected for high yield to start with. Next, please. I think this is my last slide before the acknowledgement slide, that the value proposition of the gene editing is affordable to public and small private entities, can meet pro-poor needs, crop, trade, farmers, and consumer. It can use to mitigate climate change and to support sustainable farmings, and actually also create more diversities, because you actually got a more different alleles too. And it can reduce the breeding time by at least around 2/3, and reduce the linkage drag, allowing more utilization of the gene bank material. It is very rare now, the gene bank material is being used to the mainstream breeding. And then it is faster than transgenic technology because deregulation part, and relatively accessible. Next, please. I think, thank you, terima kasih. I just want to quote Mandela now, that it's always seems impossible until it's done. With the transgenic, so I think Jalaluddin Rumi said that patience is a virtue, but it takes many, many years with the transgenic technology. But so we do have a hope that this gene editing would make it it's faster. Next, please. Oops, there's a change here. So this is my team, a picture from a few years ago, actually, at IRI, my team who work on gene editings. And this is also the Healthier Rice team. And next, please. I wish to acknowledge many collaborators from Minnesota, Dan Foitas, Thomas Chermax, he's not longer in Minnesota, Bing Yang from Iowa, Wolf from our team, and Ella Taco from Cornell University, Evan Lagunda from CSIRO, and of course all my team and countless students from Philippines, Indonesia, India, Vietnam, Myanmar, China, and USA. And this work of gene editing are co-funded by Bill Melinda Gates Foundation, Harvard Plus, USAID, Global Rice Science Partnership, in the earlier work, Indian Council of Agricultural Science and Department of Science and Technology in the Philippines. And of course, we do hope Indonesia also we will have a close collaboration in this area. Thank you for the opportunity. I'm looking forward if there's any question in here. Time is yours. Thank you.
Thank you, Dr. Ines, for a nice presentation. Since we still have 2 speakers, so I will a bit strict to the time. So the next is Professor Gede Putu Wirawan, MSc PhD, that he will presenting about, this is very interesting as well, medicinal plants from Bali Island, genotyping, bioactive compounds, and commercialization. So first, I would like to introduce Professor Wirawan. So Professor Insinyur I Gede Putu Wirawan, MSc PhD. He's a head of Central Lab for Genetic Resources and Molecular Biology at Udayana University in Bali, Indonesia, since 2013 until now, and also a Vice Rector for Planning, Cooperation, and Information Affairs of Udayana University, Bali, Indonesia, from 2002 until 2009, and a Vice Rector for Student Affairs of Udayana University in Bali, 2009 until 2013. He obtained a MSc and PhD from Graduate School of Bioagriculture at Nagoya University, Japan, and the BSc at Udayana Pak ya? University Bali. And honors and awards that he obtained is award from Minister of Foreign Affairs of Japan in 2017, visiting professor at Yamaguchi University 2018 until 2022, and Nagoya University, Japan 2019 and 2023. Indonesian-based researcher from Minister of Research and Technology of Indonesia at 2006, and Indonesia-based professor from Minister of National Education of Indonesia at 2005. His research interest is generally in biotechnology and molecular biology. Please, Professor Wirawan, the time is yours.
Thank you, the moderators, and thank you very much. I would like to express my thankfulness to the committee. And today I would like to talk on Bali, the biodiversity of Bali. And my talk is medicinal plant from Bali and the opportunity for the economic potential. So the biodiversity and nature is very much important for Balinese. So people in Bali really respect the nature as seen in the daily life of Balinese, and also for some religious ceremony. That's why I put also in my all of my product that derive from nature, from this biodiversity is inspired by nature. Next slide, please. So in terms of community, biodiversity is always related to the tradition and also to the culture and local wisdom, and very close to the biodiversity, especially plant biodiversity. One of the biodiversity is medicinal plants. The medicinal plants since ancient times were used in Bali, as mentioned in the many ancient manuscripts like lontar, lontar usada means medicine, and lontar taru permana taruh permanam in medicinal plants, and others ancient manuscripts. So however, the society used these medicinal plants without enough scientific proof. So we would prove it scientifically. So what to do? What we do, what we have done. So first we do a genotyping to confirm that the species or the variety is unique in Bali. So this Bali unique local plants, because we use wild type species of the medicinal plants. And then we analyze the petrochemical, phytochemicals, antioxidants, and others medicine properties. And then we do the functional analysis, preclinical tests, and clinical tests. And then when the time is come, we try to collaborate with the company, and with company, the formulation and regulation, and also commercialization with the company. Next slide, please. So, as mentioned previously, in current trends using herbal medicine and various herbal products are very popular in the modern society. So in Bali, as a tourism resort, as a tourism area, the tourism industry is a good market, good opportunity for herbal medicine, herbal products. Many have used the herbal products because in this case we use biodiversity for economic purpose. Of course, in our study, we produce some publications, books, patents, and others scientifically. Next slide, please. So here is the some medicinal plant that we studied. I can explain not one by one, but some. And then this seaweed. This is bulung sangu and bulung boni. This is used to consume by the community in Denpasar, in southern of Denpasar. And this seaweed is wild seaweed, not cultivated. And people in Denpasar, especially in southern Denpasar, consume this from traditionally from a long time ago. And they grow in seashore in the south of Denpasar. And also we use purnajiwa. We have two kinds of purnajiwa. In Bali called purnajiwa, and in Java they call the pranajiwa, pronojiwa. And they actually different species. And next slide. This is also we use some others. Osimum, many type of osimum. Kemangi, we call it here in Indonesia, we call selasi, kecarum, tulasi, ruku-ruku, and others. And also jeruju we use. This is also, in ancient time, they used this one and used for many purposes. Next slide, please. So, this is the genotyping of bulung sangu and bulung boni. So, bulung sangu is belong to Glacilaria gracilis, and unique in Bali. So we claim that this bulung sangu is a wild species, grow in Bali and unique in Bali. And also bulung boni is belong to Caulerva cylindraceae, and also unique in Bali. And we claim also then the sequence, the DNA sequence, the unique DNA sequence, we also registered in gene bank. So by that way we can claim that we use native Bali plant. Next slide, please. The bioactive substance function and product derived from red algae bulung sangu, Glaciaria gracilis. We call Pariastas par Serangan because it's native in Serangan. Bulung sangu contain hexadecanoic acid and hexadecanoic acid and some other chemicals, and also the carotenoid astaxanthin. Astaxanthin is widely used and commercially and strong antioxidant. So we try to use this antioxidant as a cream for skin care something, and we try also to check the anti-inflammatory content of this bulung sangu. So we found that using the mice skin, we can differentiate each other. So the control many spot of the inflammation, and then the treatment using the cream with bulung sangu extract, ethanolic extract, still normal and how to say, good without without lesion of the skin. And we try to, this is just a prototype of the product, and we use to make some cream, and then we try to use by the, how to say, voluntary use, and then we cooperate then with the company. Next slide, please. This is the genotyping of Purnajiwa, Pranajiwa. So one is belong to Kopsia arborea, and the others is belong to Euchresta onspildii. So this is Euchresta onspildii is a wild species we found here in the mountain area. And the Kopsia arborea, we found some in the lowland, here in around Denpasar. And we studied these two species. Next slide, please. So this is the pathochemical compounds of Purnajiwa, Kopsia arborea. And interestingly, this plant contains major compound is copsinin, fincadiformin. Someone said erectin, because this from ancient time this plant is used for aphrodisiac, for fertility purpose. And then we studied of this function of this extract, ethanolic extract of this Kopsia arborea. Next slide, please. So, first slide, please. Purnajiwa, Kopsia arborea, can increase the testosterone level. So, if we compare to control, positive control, sildenafil is active compound of Viagra. So it's quite similar, non-significant to the treatment of extract of Kopsia arborea. So that means this is potential use as fertility product and fertility medicine. And also they can improve the mount and mating of the, we use mice as experiment. So that means Kopsia arborea contain an aphrodisiac compound. Next slide, please. So we also make some tests of acute and chronic toxicity of Purnajiwa leaf. Why? Because Purnajiwa also used as a tea, the leaf of. So, and it is good for immunomodulator. And the result of the test of toxicity is the leaf of Purnajiwa is not toxic. So that means we can consume, use as a tea for the leaf of Purnajiwa. Next slide, please. So I produce some product. It's just for entertainment product for me. So if you come to the spa in Bali, some of them use this. The first class spa in Bali use this as a scrub, as a, what to say, cream for spa. We use tea also. But the above one, the herbal medicine, is still under clinical tests. So not produced commercially yet. Next slide, please. Okay, I come to the closing remarks. So that's a very short presentation for introduction, introducing the Bali biodiversity, Bali medicinal plant that we have studied, and some we continue to study it for the medical properties like clinical tests and others, chemical tests and some. So several Balinese medicinal plants we have studied so far show the scientific evidence, while others require some clinical tests and other tests, other research, other study again. So we've developed also some scientific product, including publication, books, patent, and others. So collaboration with company for commercialization downstream products. And we thank you for our staff, our researcher like Amy, Dara we call it, Maria Malida Fernandez Sasadara, is Sarah Adrianja, Eka Pasmidi Ariati. Thank you.
Thank you, Prof. Birawan. Very interesting topic as well. And we hold for a while for the Q&A session. We still have one more speaker. So I will invite Mr. Eilot Lam from PT Hartek, that he will present about high throughput microbe cultures for screening, media optimization, and process development. And I will mention about the CV of Mr. Eilot Lam. He's a field application specialist at Beckman Culture Life Sciences, responsible for overseeing all aspects of the bioreactor or microbioreactor, such as training, demonstration, and troubleshooting. And he's now at PT Hartek, yeah. And educational background, he obtained a Master of Science in Molecular Biotechnology in Hong Kong, and the BSc in microbiology in Australia. The specialization is life sciences, medical devices, and molecular biology. To Mr. Eilot Lam, yes, the floor is yours.
one? So, yes, yeah, ready to go.
Sebagian di Jepang, di Kyoto, sebagian awalnya di sini, gitu. Punya siapa di polisi, di lab toxicology and forensic. Besar, alatnya baru-baru, bagus-bagus. Aku sana, oh ini bagus-bagus alatnya, gitu. Itu umumnya kan digunakan untuk forensik ya. Tapi apa namanya, kalangan universitas banyak sana. Bagus kok anunya, hasilnya itu. Jadi berapa kali kita ulang gitu. Jadi sekali itu udah coba ulang lagi gitu. Kemudian di confirm di Kyoto juga, oke, gitu. Pak Master masih kan di tempatnya semula di BB Biogin, tapi BB Biogin berubah nama sekarang gitu kan? Oh gitu.
Oh iya, bukan ya, aku nyambil.
Dan selesai kan Pak Bambang tuh.
Oh ada cookiesnya. Ini dia soalnya pakai laptopnya sendiri, jadi agak lama settingnya. Dia baru datangnya malam.
Okay, morning everyone. Sorry for the waiting. So it is good to having a time with expertise working on gene editing. So now I'm from Batman Kota, knife science. So my name is Ilot. I'm talking about the bioreactor or fermenter. Okay, so then I go straight to the hardware. This is the Biodata XT. Okay, and then you can see from the top there is a blue cover, so you can put the plate in inside, and it is where you perform the bio process. Okay, so underneath the incubation chamber, there is some laser to do the measurement, well to well, row to row, and column to column. Okay, so from the front, you can see this slide door, a little bit transparent. So what it does, you simply put a 500 ml water into it, the deionized water, and then it is for the humidifier to maintain the humidity throughout the process. Okay, so, and also you can, on the top of the hardware, the device, you can see the monitor. So you actually can, which is touch screen, you can do some adjustment and to initiate the protocol that you have already done. Okay, so with this monitor, you can stop, pause, and stop the experiments, and also you can do the adjustments such as temperature, the shaking speed, and also the gas load and gas concentration. Okay, so this is interface, so you can adjust it in real time, as this device provides you the real-time result. So after 1 or 2 hours, if you don't like the speed, if you don't like temperature, you can increase it up or down. Okay, so and then you open up this cover. It's for you to put a sensor. We call this filter, for you to collect your information or the data during the time. You can switch it. It is easy to done by 2 minutes. Okay. Right. Okay, so we have the upgraded version for this XT. We just connected with the, we call this valve control units. With this unit, you're able to doing a fed batch or a pH control. Okay, so I will go for detail in a minute. So when you compare, economic compare, from the cell engineering to the final products, okay, though it involves a lot of steps. You have your candidate, bacteria or microorganism, then you have culture it, and then with a bigger flask with different external device such as pH meter or thermometer, and then go for the large scale. Okay, so you need to do the screening optimization, and then develop the process, and then finally you scale up and then get the final products. So you can see the working volume which is increasing. So for those steps, we call upstream steps, so it involves manpower time. So why don't we just scale down a bit and then work on it easily and more efficiently. So we need a device that can able to do it. Okay, when compared with conventional technology, from left-hand side we have a standard size of fermenter. The right-hand side, each time the operator can handle 8 or even 10, depends on operator. The right-hand side, the big size of bioreactor, each time may be handled 2 or 1 because of the volume or how you handling the device. For the XT, each time you can conduct 40x process. And also, with conventional ones, you need to connect it into external devices and also some control panel, etc. So for the XT, you need a laptop. We have a software for you to develop the protocol and also to analyze the result. If you decided to use study fed batch, then you need one more device, which is called valve control units. Let's keep going back. Okay. So and the scalability, which is 20,000. So that means what happening in the XT is reflecting what will happen when you go into full size, big size. We have a good paper by comparing different systems. Okay, so we have XT 1 mil and then 1 liter, and the biggest size is 20 liter. And this is the result that sorry about that, okay. So the right dot, which is glucose consuming, it is going to consume all the glucose around after 13 hours. And then the cell dry rate, which the bacteria is entering the stationary phase after 12 hours. And a similar pattern for the enzyme activity curve. So when you compare those three systems, they're showing the same trend and same pattern. Okay, so for example, it is the well we're going to conduct the experiments. If I'm using 1 well, which is 1 mil equal to 1 flask, okay, 1 bioreaction, plus it can as function as data connection, data entry and shaking, and also it can adjust temperature. So if I use all the plates, which is 40x, it is equal to 4 sets of incubator or shaker with 12 flasks on it. So you can see we can save some medium the preparation time and reduce data connection time. Okay, so I'm going back to the play again, and as we can play with different parameter, with pH medium, which type of candidate, the microorganism, and also feeding strategy or the medium. Okay, so this is the plates I put different medium in different role. Okay, so with different pH gradient, from lower pH to high pH. And then using same microorganism. Then I can screen out in which pH range this equalizer work best for which medium. Okay, and alternatively, I can put one medium, such as glucose, but I use different, as well, using different pH range, and then but different microorganism. And again, I can screen out which microorganism doing best for this glucose medium. Okay, and then I also can, I would say the second step, or I very good know about which bacteria work best for which medium, and then I can do this arrangement. Or I can split the plate into two, and then also with different concentration of the medium. But I can set it in different pH. The left-hand side from no pH to high pH, and the right-hand side high to no. Then it's easy for me to screen out which pH range, which medium concentration with bacteria work best, and I can get what I wanted. Okay, so this is the result. After the experiment, you can analyze, compare, and display for the exact set of data. For example, it is a curve for the biomass versus time after 65 hours. So it can easy to point out which one is negative and which organism doing the best get the higher biomass. Okay, the feature. Okay, the XT, the temperature, the incubation temperature up to 50 degrees Celsius, and the check enzyme, the maximum, which is you can opt to set different of the checking speed in terms of RPM. And then we can put 6 different filters into it, such as the filter to measure pH, dissolve oxygen, or even green fluorescent protein, etc., etc. And we have a software for you to develop the protocol and also doing the analysis after that. We measure biomass, we do the pH measurement, DO measurement, which is invasive, without disturbing the process. Okay, and for sure, the last one, dehumidify, to maintain the humidity throughout the whole process, around 80%. Sorry, keep going back. Okay, so if you decided to use the valve control unit, you can perform the fat batch mode. So you have add up 2 more features, the pH control and also the fat batch. Okay, a couple of months ago, we launched a new module, which is called LAM module. With this module, you need 2 devices. The first device and the second device. You cannot get the second device with basically having the LED light from the bottom. The range, which is 365 to 850 nanometer. With this module, you can cultivate. Sorry about that. You can cultivate microalgae and cyanobacteria, those organisms that can conduct photosynthesis. Okay, so, and also gassing module, when we studying microorganisms, we may encounter aerobic, anaerobic, and microalveolic. And then we have different gassing module for particular study. So for the default module, we use in the air from outside the atmospheric air. And then with an aerobic study, we're using nitrogen gas to flux the system to remove all the oxygen inside. And for CO2 upregulation, it is up to 12% of CO2. And the O2 downregulation, 1 to 21. It should mostly cover most of the oxygen demanding microorganism. And lastly, the oxygen, the O2 up, which is you can go for extreme, 100% of oxygen. Okay, so those are the optional gassing module. Okay, the micro-data plates. Okay, so this is micro-data plate you're going to put into the XT and perform your study. I'm going to play the video to see if it works. Okay, so in build situation is put it into biosafety cabinets. Can you play the video?
Potential penetration of solutions into microchannels. And manually apply the sterile barrier thread, which seals the wells off from the environment. Be sure to apply the filter properly and to attach overlapping edges to the sides of the MTP. In the next step, apply the silicone foil atop the sterile barrier foil with the help of the foil applicator XT. It is recommended to sterilize this tool prior to use by autoclaving or webbing with alcohol. Place the silicone foil inside the foil applicator with the brown protective film facing upward and press it down so that the pins push through the corresponding holes in the foil. Carefully remove the protective film. Place the construct atop the MTP and press it down firmly. Now, last step, place the gassing lid on the MTP and push it down so that it sits in the correct position.
Okay, clean and tidy. Thank you. Okay, so we have a microfluidic plate, as I mentioned, which is for fat batch and the pH control. Okay, so this is a microfluidic plate. Underneath the plate, you can see the micro channel. Okay, for the top, you can see the first two rows, which is occupied with either feeding medium or the pH buffer, sodium hydroxide or hydrochloric acid, base and acid. So you can occupy them with okay. So for example, you can put glucose in the first row and then sucrose in the second row, or acid and base. But most of the time, we put medium, and also the base should be more than enough. So when you look through the plates, you can see this is the arrangement. I'm going to enlarge a bit. Okay, so I'm going to bomb the second reservoir into my cultivation well. I need to bomb the sodium hydroxide into it, and then it's going to follow a particular path without disturbing the other cultivation well. And the situation is similar to the this well. If I want to bomb the feeding into the cultivation well, and again, it's going to burn through a particular individual microchannel path. So for this technology, we can address different pH in different cultivation well. For example, this one 5.5, and then 6, and then so on. Okay, I'm going to play a short video again and see how the mechanism works. Yeah, this is the video. Okay. Okay, so after the experiment, you can get the result. This is the interface. We call this BioLab, the software. You can see the biomass, pH, and the dissolved oxygen. You can click on each of that panel, and then you select which parameter you want to focus on. And also you focus on which well. In this case, I just select all the well. Okay, so I'm about to finish my presentation. Okay, so before finish, I have some information. You go to Google and type in BioLab video, and then you click on those links. The first or second, they will do. Okay, so you go to the landing page from Batman Coulter, and then it's specific for BioLab XT. You can check on those videos, including software, the hardware setups. Okay, so just Google it, BioLab XT, and then video. Okay, so thanks for the time. It's the end of presentation.
Thank you, Mr. Elatlan, for the nice presentation. Is it, or you still have presentation, or it's just finished? Okay. All right, ladies and gentlemen, we all have wrapped up all the 5 speakers this morning. So now we are turned into the Q&A sessions. I already have actually 2 questions from online, but I will give first for the offline questions. So for the first time, I'll give you the opportunity for 3 questions. Please. You can mention the question to which whom the speakers and what's the questions. Yes, just mention your name also first. Okay. Second one, if there is from this row or this row. Yeah. Okay. Silakan Ibu. You can start the question.
Thank you, I'm Astri. I would like to ask about to Prof. Gede, because I'm starting to study about the ethnobotany on my research now. So I'm really interesting because in Bali, the people still, many people still use the plant for the medicine, ya Pak. My study is in Kalimantan, so it's not a tourism place to, I hope the people in Kalimantan also can use the plant for increase their economic aspect. But I still thinking how do you think if we use the plant as medicine without exploitation the plant also like that. Also, do you have any criteria about the plant that can be explored to be a commercial thing? Thank you, Pak.
Will you mention your name, please, Ibu?
I'm Astri from Universitas Jenderal Sudirman.
Thank you. Shall we collect first the question, or Professor Wirawan will directly answer. I think Professor Wirawan, please just directly answer it.
Thank you, Ibu Astri. How to avoid the exploration of the wild plant especially in Kalimantan for medicinal use? So difficult to answer. So one thing, maybe a common thing that I can give is cultivate the plant by nursery and traditional nursery, and also nowadays we can use tissue culture or something. So for us in Bali, for me in Bali, like Euchresta, Honspedi, Purnajiwa, very rare already now in the mountain. So we try to cultivate them by tissue culture. So then, I don't know, maybe the compound also, the content of the compound become changed because some gene expression changed with the different media and something. But if we culture again, bring to the forest, to their habitat in the mountain, I think that will be back again to the normal plants, to the what we get the phytochemical previously. That's one thing. But if we make a nursery by cultivating their seed, maybe their seed using polybag something, we can get the plant, but it takes time also. And then we use the plants is depend on the how to say, depend on demand of the customers. Depend on the demand of the customers. So if it come to the many demand, is this problem also. So if in Western country, like also in Japan, they get the chemical from the plants, and then they make artificially the chemical, and then they use as medicine, for example. But here in Indonesia, in Bali, the people love to use herbal. So why don't we create some, increase some herbal medicine by using some analysis, scientific analysis, and then also the cultivation of the plants. That's Ibu. Thank you.
Okay, thank you, Professor Wirawan. Professor Annie, or maybe we'll add some comments about it.
Yes, please. Bu Astri, Bu Aci, thank you for the question. Although the question is for Prof. Wirawan, but I would like to add some information from my talk. In my talk, I already mentioned that metabolomic is based in the biology, system biology, because it's affected by environment. So as Professor Wirawan already mentioned, that it might be affected by the environment where they are grown. So that's why from my research, my team research, it's affected also from the by the artificial light. Yeah, so I didn't explain into details this morning, because of the time. But definitely certain lights, either the one that I tried for one variety, affected by red light, for example, but it really depends on which part of the plant, the organ. So although we need to check further whether the effect is stable. Yeah, so that's why it's really affected. I mean, the content of the compounds or the metabolite. That's why we did the integrated omics approach, because from the RNA transcriptomic, where the trait is expressed, and also from the DNA, so we can do the genotyping. So then with the metabolite, for the metabolite. So that's why it's really critical which media or which environment they are grown, even under tissue culture condition. Yeah, thank you.
Thank you, Prof. Annie. Other questions or comments from the floor for all the speakers? Yes, please. Please mention your name and institutions. Thank you.
Good morning. My name is Nadira. I'm from Merck Life Science. My question is maybe for both Dr. Rongdi and Dr. Ines. So during my master's study, I had the chance to study and did small experiment with CRISPR. And although it's an animal, I have witnessed the first-hand of how CRISPR is very powerful. And I still believe that it's one of the most potent solution to aid the food security. So it's CRISPR for food. But as you may know, and maybe you have got these questions a lot, when it comes to gene-edited plants and food, we're still so much burdened with the public perspective on GMO, the term of GMO itself. So the term of GMO scares people still, and unfortunately, how do you think as a researcher we can educate the public that GMO plants are actually safe to be consumed? Okay, that's all my question. Thank you.
Okay, thank you. I think you can already start the answer.
Okay, I'll answer this one first, then I can turn it to Ines to answer you. We, at Rutgers, we also have this major we call the biotechnology major, and we have the class for freshmen and also or sophomore. It's a required course. It's called the ethics and issues in biotechnology. So we do teach that. So I think very important is the education. As a scientist and also as science major students or graduate students here or anywhere, that we have the duty to educate the public. So I started with my family. My parents were civil engineers. They had no idea about plants. So I talked to them, and I talked to my uncles and aunts. They're all in China, or cousins. And if we have the WeChat group, if they post anything, say this tomato looks weird, it must be GMO, and I would tell them. And so in our class at Rutgers, and we make the students discuss what is a GMO. And normally my slides comes, what is a GMO? How do we make a GMO? What do we need to be concerned about GMO? As all scientists here, we all know they are very safe, and especially for just making even transgenic. Or when you make gene editing, you can make I showed you the diagram, the guide RNA and the Cas9 that are in a plasmid. What is a plasmid? So I start telling them, we make these genes, it's part of, it's a gene. Genes it's made up of DNA. DNA consists of ACGT4 nucleotides. It's one gene among any other genes. So it's the same composition, and it's all your other genes. It's in human, it's in insects or pigs or the plants that we engineer with. So for example, rice or sweet basil, we make this construct, it's just one gene, all of millions and millions of them. That's one. And two, is GMO safe? So like I said during my talk, even us, we're GMO. We are crosses between mom and dad. And the plants, whatever we eat without the breeding, and during, I normally start with traditional breeding, we are moving actually thousands and thousands of genes from one cultivar to the other. And now we're doing GMO, we're moving one gene. Number three, I tell them, now we engineer the plants. We're eating the plants. For example, Dr. Ina's rice, we eat rice every day. So it's DNA. Once a human ingests the rice, GMO rice, and that DNA is chopped into pieces, just as all the other millions of DNAs. Fourth, I tell them, those genes that we engineered the rice with, they contain plant promoters. And they do not work in your tummy, even if the DNA is not chopped into pieces. So what are you worried about? That's how I normally start with the most simple science, and then explain to them if they really want to ask more, and I will say how I make them, and the process that we make them. So, and I said, often, if you do want to worry about one thing about GMO, it's, for example, the traditional way that we have a selectable marker, and often it's antibiotic-resistant gene. And then when the second people hear about antibiotic, and they say, okay, we're all gonna die because once we have bacterial, fungus infection, we are antibiotic resistant. So I said, then go back. I told you, this is one gene. It will be chopped into pieces in your tummy, and it's expressed by the plant promoter that will never work in your stomach. So that's how I normally start. And I can give you one example. As I was invited to hold a public forum in New Jersey, in United States. So I told them about what is a GMO, how I make a GMO, what are the things that you need to be worried about. Yes, of course, there are other genes I will be worried about. For example, I engineer turf grasses, and they want herbicide resistance. So I told the company, I can give you two ways of making the herbicide-resistant turf grass. One is totally, I don't need to use any transgene, no foreign DNA. I put the herbicides in the media, and I make them screened, even from the callus, and then I screen them. And most of them will be killed. And then I regenerate those that survived the selection, and then the herbicide in the media force them to mutate. Most herbicides are very specific herbicides. They target only one gene. So then the mutation is made without any transgene. Do you want this type, or you want, I can make the transgene and I will make them herbicide resistant. Of course, when I hear that I can make it just by tissue culture methods, so I'll try this one first. No problem, we can do that, right? So then if you do make the transgenic herbicide resistant turf grass, it's not allowed to be released into the wild because most grasses are cross-pollinated, and this resistant gene can spread out to the others. That's why they're not approved, right? So we can do the experiments, and we can prove our theories, we can prove whatever we put in, it should work. But we need to have those concerns. So I will tell the public what I'm concerned about, what I'm not concerned about. For food, most of them, you should not be concerned about it at all. So that's how I normally start. So in that public forum, after I explain all of this, and there's someone that came over to me and saying, okay, Dr. D, if I put a glass of glyphosate, the herbicide, in front of you, would you drink them? And I say, definitely not. And her idea was, if I engineer the plant to be glyphosate resistant, and we're gonna have glyphosate everywhere, and she's gonna drink them because they leached into the soil, into the water, and then she's gonna end up drinking lots of the herbicides, right? So I told her, definitely not. And so I told her again, how does it can be made? And after my explanation and everything, then she came over to me, she said, well, thank you very much, and you really answered my question. So with this example, I think very importantly, we need to educate. If they are not in the science field, if they are not, they did not study biology, like my parents, I would tell them how it's done in the simplest term. And then tell them what I'm worried about, what I'm not worried about. For the things that I'm worried about, how I can take care of it, how I can use other technology to solve it. So for example, I made many different kind of transgenic plants, including transgenic rice, I told Ines. And, but now that's why I wanted to use the gene editing, because there's the possibility I can make them without the transgene, and then just to relieve people's concern. But still, education is a key. We need to be open, and we will let people know how it's done and what are the benefits to the environment, to human health, and to the world.
Okay, thank you, Dr. Rongdi. Okay, Dr. Nancy. Maybe just.
Thank you. I think Dr. Rongdi has already given a very clear explanation. I just want to add few things. I think we also need to separate when we did talk about GMO and gene editing, because otherwise gene editing would end it as the GMO in the past. But education is very important. When I talked about GMO, I also similar to that, I will make a simple analogy. Like we humans have 233 bacterial genes. So while I'm talking to you, bacteria maybe try to move their genes into my to my body. And I once was in Australia, and this taxi driver was so scared about GMO. And then I said, I put gene from apple and from rice to rice. And it's actually the protein produced from apple. So if you add apple, you already eat that protein product. And then he said, well, I always heard that GMO can transfer the gene into us. And so we are become mutant. I said, well, if you are that easy, by now you are already walking green, because you eat the spinach, you eat this, you will be Hulk by now. And he was laughing, but they said, okay. Then eventually it was like in the lighter, and then say, oh, it was the same product, yeah. So that sometimes we need to show, but it's also important to show that GMO has gone through so many regulatory pathway, and give example of the country that people think is very careful, like Japan. They have approval. So many events are approved for consumption in Japan. So this sometimes, oh, okay, that's different. But then we have to, when we talk about gene editing, we need to clearly separate it from GMO, because of this stigma. So we say the editing is actually variant, and this variant happen all the time. You eat in nature, because when we eat aromatic rice, is edited by nature. When we in rice itself, it's one single nucleate every 18 base pair. So imagine, so nature always editing all the time. So when we clearly said it is because sometimes even mutant, people are afraid of mutant. So we tend to say, oh, the variant, but some people don't understand variant. But it is like separate gene editing, because one can actually cannot differ in the product itself, whether it is obtained through nature or obtained through editing. Because the entire modern rice variety is based on mutation on the semi-dwarf gene. So it is also just like giving a simple example. One can win, one cannot win, but education is very important. I also give example, for example, in one of the Indonesian book for, I don't know how it went to the education book about GMO, and it said about like GMO is a very dangerous, and it was in actually buku SD atau SMP. So I think all the people in the university also is responsible to check all these things. Do not go to, because whoever speak to you first is often make a difference. If somebody already brainwash you, this is something dangerous, it might stay. So who's informed, because we did a lot of survey, who they listen it first from does make a difference. So I think that is also responsibility of the educator. Thank you.
Okay, thank you, Dr. Ines. Yes, this still challenge about the public perceptions, yeah, between the GMO. Maybe short questions, sir? Okay.
Thank you. I just want to make short comment regarding this issue. I think this is just a matter of priority. I'm a scientist, but from academicians, but I am also managers. If you're a manager, just, of course we need both. If you ask, you need more food or more help? If you ask that question to United States people, maybe different if you ask to the African people. So just a priority. You need more health or you need more food? That's one.
Okay, thank you. Yeah, the question is who will set up the priority? Okay, thank you. I'm sorry because of the limited of time, so we will move now to the online questions. So the first question is to Professor Annie. The question is, why mushroom grow on elephant dung is interesting to study it further. Is there any indication that the mushroom contain important biochemical? You will answer directly.
Yes, definitely. We have a research team at BRIN, well, before LIPI, that the result was a lot of compounds found from mushroom, in mushroom. Yeah, so we have collections of mushroom quite a lot, a number of, a set of collection. And a lot of compounds actually related to health also. If I was given a bit of time, I can look at my note, what kind of compound that they have. But that definitely, I mean, even for the Japanese, for the Chinese also, yeah, we have like shiitake and other mushroom. But Indonesia, we have also a lot of mushroom that I also presented on my slide, the morel mushroom, but in the elephant dung, I need to be characterized more to get the exact what the species, for example, like taxonomical study and also the content from the elephant dung. But definitely, if you use even only the dung, yeah, like a dung of well, from the cow or from goat or from chicken, poultry, that's also we use as an organic fertilizer. So definitely from the dung, it's we can extract something, and also the mushroom that grow in the dung. Like in Thailand, they use quite a lot of elephant dung for fertilizing their garden or their plants, horticulture or food crops. So this one, yeah, I will later on give you the series of content of the compound in the mushroom, yeah. So, but this is interesting. I mean, mushroom and microorganism not really been explored as much as animal or plant. So that's why when it was sleepy that time, before it's transferred to brain, we publish a book or a publication that we found plant first, so that's why new species found in the wild in Indonesia. The plant, new species of plants or flora are higher, but then animal first or fauna and then plants, and then very low number of new species of microorganisms. It's not because they not really have high diversity, but it's just the number of research or detailed research or intensive research on microorganisms not yet done. But then after that, since like 10 years ago, I mean the research on microorganism, this can be like macro fungi or micro and macro that can be also explored more. And I'm sure they have a lot of potential on compounds, active compound or compound related to health. Yeah, thank you.
Thank you, Prof. Eni. Still have a question to Dr. Rongdi. Is there any best way in controlling fusarium disease on banana and papaya, since it is a lot happens in Nusa Tenggara Timur? Yeah. And the second one to Dr. Rongdi is a factor for CRISPR translation. Is it different than the one that you use to transfer into the genome? Thank you.
Thank you for the question. First one on fusarium, and they're all fusarium, of course, different species. So in wheat and barley, and sometimes on maize, even on maize, we have other species. So on barley and wheat, they are fusarium graminarium. On banana, it's a different one, but they're all fusarium. So fusarium is considered as hemibiotrophic. That means they started as biotrophic fungus. So that's when I call it intimately interacting with the plants. So that's why we can engineer the host. If we know which factors that they really do intimately interact with the plants. As of now, we don't know any. That's why the genes, so there's still a lot more that we need to do, right? So the genes that I choose so far, one is called the 2OGO, the other one is the HSK. Even with the HSK encoding homoserine kinase, we still don't know how exactly it works. So people have put homoserine, because if you knock out the homoserine kinase, the homoserine content went up in the plants. And then they just put the pure homoserine onto the media plates, and then they found out that can actually inhibit the spores germination and the spore development. So that may be because of this, and that's how they kill the fungus. So this type of host factors we can engineer so far. So after the initial infection by fusarium, let's say graminarium, into wheat and barley, then they release this toxin, this fungal toxin or mycotoxin. Mycotoxin kills the cells. Once the cells are dead, the fungus of fusarium goes more like a saprophytic fungus. We all know saprophytic fungi, they make use of the dead tissue, then they go wild. So that's why the infection goes even further. So that's why if to engineer plants, you need to know what does this fungus interact with. If you don't have the specific factor yet, you can engineer other, we call the negative regulators in the host immunity. So if they're negative, you cut that out, then the plant goes to the positive way, which is disease resistant, and that's what we want, right? So if there are other diseases, for example, rust, powdery mildew, downy mildew, and those are obligate parasitic fungi, they have to interact with the host very, very specifically. Some of them even have a host-specific genes that they interact with, and those are the great factors that you can engineer, manipulate by gene editing to knock those out. But if they're saprophytic, it will be kind of hard. So you can manipulate the negative regulators in the immunity, and that's what we're doing now with fusarium. So the question is, can we do the same for banana fusarium? Definitely, yes. And because if it's a fungus, it's in the same genus, they interact with whatever the host a bit similarly. So you can always work on that. And so that's the first question. My second question is that the transient vector to engineer plants versus the integrating vector. Is there any difference? Whether it works or not, they're all vectors. So even the transient one, I call the guide RNA cassette. It has the small RNA promoter. In plants, mostly we use U6 or U3. And then it has the stop codon is 8 or 9 T's, so we'll stop that. And then the Cas9 cassette, it's under normally constitutive promoter, for example, 35 is a promoter or ubiquitin promoter, and then you have the terminator, right? So my transient vector has the guide RNA cassette, the Cas9 cassette. It just doesn't have the left border and the right border. So the left border and the right border is for the integrating vector to integrate this tDNA region into the genome. So that's the difference between those two vectors. So the transient vector is not supposed to integrate into the host genome, but they do. I have seen the first article is on wheat, and they found about 55% of all the population or individual plants that they screened, they did integrate it in. So I screened mine, basil, we screened lots of them. We found about a bit less than 50%. They did integrate in. So we choose the one that did not have the transient vector integrated in, and we analyze those. So does it work the same as the integrating vector? Of course. I showed you all the mutants that we have made. It's because whether it's transient or not, once the DNA is gene gun bombarded into the tissue, and the vector will, the promoter will start to work, will transcribe the gene, will express the gene. And I told you, so remember, the shortest people have seen in animal cells, Cas9 will start to make mutations in as little as 15 minutes. For plants, we have tested in Arabidopsis protoplast and in sweet basil and grape. I also work with grape protoplast. I couldn't let it go on for only 4 hours. So I made 24 hours. I incubate them together 24 hours. The next day we harvest all of the cells and get the DNA and analyze them. I have mutations. So with such a short time, integrate or not, transient vector, if they don't integrate, the mutation is already done. So it's quite efficient. Yeah. Integrating vector, yes, they do integrate it in. Normally when you do agrobacterial transformation, you give them 2 to 3 days. That's allowed the agrobacterium to infect the plants. And then you will kill the aggro, then you screen your plants. By that time, if you have the CRISPR vector, the mutation is already done.
In any kind of, any species of plant?
Yeah, in most of the ones. I work with grapes, sweet basil, barley, and we have tobacco, Arabidopsis, and we have many different kind of turf grass, and we have checked them in like 24 to 48 hours. The mutation is already there.
Very
so it's very efficient.
Okay, thank you, Dr. Rongdi. Also, we have 2 questions for Dr. Ines. The first is, I would like to know how is the mechanism so there is no transgene or scissors in GE events. How can we increase the number of events without transgene or the scissors? The second one, how is the progress and update of the transgenic rice of improved rice with beta-carotene? I knew that the last time it was promoted in Africa. Is there any other plan of kind of transgenic rice will be launched by IRI in Philippines? So maybe Dr. Ines can also answer it in more concise or short time.
The first question I heard very well, but the second not really. But I answered the first one first. Okay. I think in the case of rice, then what we do is we segregate it out So it was a stable integration and then segregate out in the second generation. It's like a Mendelian segregation on the two different locus. And our mutation efficiency is very high, between 60 to 80% is edited, the line are edited, then you segregate out in the next generation, basically. But there are techniques now, as mentioned, I think it's very cool what Dr. Rondi explained, but there are now also other techniques for non-DNA integration, either by viral infection, and also actually kind of you introduce it like a suicide, something that on the actual, the other one, those who has still the scissor won't grow further. But it would take a long time to explain it, but I think basically for cereal is easy, you can segregate out, but if you work with banana, clonally propagated plants, then it's a different story.
Okay.
What was the second question?
Yeah, actually it's about how the progress about the rise, transgenic rise of beta carotene in IRI.
Golden
It's
rice.
the last time it was promoted also in Africa. So
No,
is there any other plan of kind of transgenic rice will be launched by IRI?
For the golden rice in the Philippines, it has got already commercial approval, and there has been 2 season on seed production of it, and so it's including in fact if for like children feeding for in school, you get certain days. But there is a challenge recently with what they called for both BT eggplant and transgenic. The Supreme Court released this writ of kalikasan, which is meaning that it may have environmental effect, but Department of Agriculture has countered it. So we'll see how it goes, but this is working on transgenic is always challenging, but on the other hand, there's a lot of support, and the farmers is actually asked, how can we get the seed, and then the second transgenic product, as I mentioned in my talk, would be iron and zinc rice, which is going to be pyramided with the golden rice. And then we also work on photosynthesis. This is with EU funding, so it's to improve photosynthesis.
Okay. Thank you, Dr. Ines. So, ladies and gentlemen, unfortunately, because of the time limitation, and we are running out already a long way from the schedule, so we have to close the question and answer session. And please give a big applause for all the speakers again for a nice presentation. Okay. And thank you for your attention. So I will close this session. I will give it back to the Master of Ceremony. Thank you very much.
Well, thank you very much, Dr. Hayati, for chairing the plenary session. And we would like to thank to our invited speakers for amazing presentations. Their topic must be very fruitful for our future academic journey. We would like to present certificate of appreciation to our invited speakers. Therefore, to our invited speakers, please come forward in front of the stage. And we would like to invite the representative of our sponsors and also from IBC to CropLife, Corteva, Merck, Professor Bambang Prasetyo, and Mr. Engineer Master MSc PhD to give certificate of appreciation towards our invited speakers. Token of appreciation to our invited speakers. So our invited speakers receive traditional Balinese merchandise. So for the male speakers, they receive udang, traditional headwear, usually used by males in Bali for cultural and traditional ceremonies. And then for the female speakers, they receive fans. It's a symbol of Balinese culture, which we often use for traditional Balinese dance. We also would like to invite all of the guests and also sponsors, invited speakers and moderator to remain on your position so we can have photo session together. There is spot left still on the left of the invited speakers. One, two, three. Say Bali in 3. 1, 2, 3, Bali. Okay. Thank you very much to our invited speakers, distinguished guests, sponsors, moderators. We kindly invite you to be seated, to return to your seat and be seated. And ladies and gentlemen, after this we will have a word of appreciation from Rutgers University to Universitas Nasional students. Well, ladies and gentlemen, we would like to invite you to be seated as we will proceed to our next agenda, which is certification award from Rutgers University to the students of Universitas Nasional. Therefore, we would like to invite the representatives from Rutgers University, Professor Ilya Raskin and Professor Slavik, to present the certificate to students from Universitas Nasional. Please to professors.
Oh yes, a couple of things.
So before we invite the students, there is a remark from Professor Ilya Raskin.
Hello, and this is truly a wonderful opportunity for me to welcome and to graduate 8 trainees which came out of the result of the funding from National Institutes of Health with the Center for Botanicals and Chronic Diseases, which is something we have started originally with Tajikistan about 8 years ago, and Indonesia joined the program about 3 years ago, and our first wonderful crop of trainees from the second round are graduating today. I cannot tell you how delighted my colleagues, Dr. Dushenkov and my American colleagues, Antonia and Isabel, are proud of our trainees. You made this conference possible with your Indonesian colleagues, and it's been a real joy to see how you mature, and moreover, we've learned from you just as much as you possibly learned from us. And we developed this very important international friendship, and hopefully it will last for many, many years to come. So without further ado, I think Dr. Dushenkov will present the diplomas. We have 4 out of 8, we have, I believe, 4 trainees who will be physically here today. The rest will get their diplomas some other way.
Right.
And of course, the head of our program here, which is CBCD, Professor, please, Irnawati Sinaga is here, and we're truly extremely grateful for her contribution to this program. She's been a wonderful colleague and a friend, and truly devoted to her students.
Thank you very much, Professor Aurelia, for your remarks. Therefore, we would like also to invite Professor Erna from Universitas Nasional to come to the front of the stage together with Professor Ilan, Professor Dushenkov, to present the award. And we would like to call the students from Universitas Nasional one by one to exclusively receive the award of appreciation from Rutgers University. Therefore, we would like to start with Ms. Hanifa Nur Adila. To Ms. Hanifa, please come forward. Congratulations. Please everyone give a round of applause to the students of Universitas Nasional. And each student can have photo session with Professors Ilya and Professor Dushenkov and also Professor Erna. Okay, thank you very much to Ms. Hanifa. Please remain on your position as we will call your friend, Ms. Novia Deliasari Alia Azahra. Congratulations, Ms. Novia. Now we will have a photo session together. Thank you very much, Ms. Novia. Now we will call the third student, Ms. Shafira Soraya. Congratulations to Ms. Shafira Soraya. Thank you very much, Ms. Shafira. Now, last but not least, we would like to call the last student to receive the award of appreciation. Congratulations to Ms. Astri Rozana Siregar. Congratulations, Ms. Astri. Now we will have a photo session together with Ms. Astri. Okay, thank you very much. Now we will invite back all of the students, Ms. Hanifa, Ms. Novia, Ms. Shafira, and Ms. Astri to take a photo session. And after this, we will call all mentors of Center for Botanicals and Chronic Diseases, Mr. Tatang and Mrs. Nonon, to also come forward to the stage. Okay. Now we invite all mentors of the Center for Botanical and Chronic Diseases first, Mr. Tatang and Mrs. Nonon, to have a photo session together. And then afterwards, we will invite all lecturers and students. Right now, we would like to invite all lecturers and students of Center for Botanicals and Chronic Diseases to come forward and take a photo session. Take a picture together in the last or final photo session. All right, is that all? Oh, to the distinguished guests also from Indonesia, we would like to invite to take a photo session together, Professor Annie. Okay, thank you very much to all the students of Universitas Nasional. Congratulations once again to the Center for Botanicals and Chronic Diseases. Thank you very much. And ladies and gentlemen, we also would like to have a photo session together with the online participants. So online participants, therefore we would like to invite our distinguished guests, invited speakers, keynote speakers to come forward. We also would like to invite first Mr. Mastur, Professor Bambang to come forward, Professor Annie, all of the keynote speakers, all of the invited speakers, to have a photo session together with our online participants. Therefore, to our online participants, please stay with us and be in front of your gadgets, and please open your camera so the committee can take a picture with all of you. To all invited speakers and keynote speakers, please come forward. Once again, we would like to make sure all participants to open their camera. All right. Okay, so 1, 2, 3. Okay. 1, 2, 3, cheers. All right, one more. Okay. The IT team will assist the online participants in taking their picture. 1, 2, 3, cheers. Okay. Which one? This screen? Di sebelah kamu sini, Bu. Biar lebih luas. To all invited speakers. Also, we would like to invite to take a picture again with our online participants over there in front of the screen. So the online participants can be seen and documented as well. There are 5 slides. 5 slides, so 5 pictures that we all should take. We will move to the next slide. So please, for the photographer, take picture with participants in slide 2. 1, 2, 3. One more. 1, 2, 3. Okay, now we move to the slide number 3 with participants on slide number 3, please open your camera. Okay, we're just going to take pictures. And then with participant slide number 4. 1, 2, 3. Great, now we move to the last slide of the participants. This is the last photo session with the online participants. 1, 2, 3. All right. Well, thank you very much. This is the first slide. We have taken a picture with these participants. Thank you very much to our guest from IBC, from one more photo session with all of the participants that are present in this room. Therefore, we would like to invite all participants that are present in this room to come forward to have a photo session together with all of the committee and guests of this international conference. Kami undang Bapak Ibu yang hadir di ruangan ini untuk maju ke depan dan berfoto bersama.
This is the proof that you are here also on the second day. Otherwise you're missing somewhere. Sightseeing.
Once again, we would like to invite all of the participants, presenters who are still in this room to come forward to have a photo session together. Our committee will arrange your position. Okay. Ini pengumuman ruangannya apakah setelah atau setelah? Okay. Have all participants? Okay, everyone's ready? Okay, 1, 2, 3. Okay, and then together say Bali. Okay, in 1, 2, 3, Bali. Okay, with brightest smile. Okay, one more. Okay, now with phones. Okay, 1, 2, 3, Bali. Okay, that's all. Okay, thank you very much to our participants, distinguished guests, keynote and invited speakers. Please kindly return to your seat. A little bit information from the committee to all participants or presenters that are available for tomorrow. If you want to register for the field trip to Botanic Garden Bedugul, please register yourself at the registration desk outside with the committee. The trip is free and the bus is ready tomorrow at 6:30 a.m. and the bus will leave at 7 a.m. So if you have available time to go with the committee and the members of this conference tomorrow to Bedugul, we would like to invite you to go as well. We also would like to thank our sponsors. Therefore, we would like to express our gratitude to our sponsors for supporting this event, and please enjoy this video of sponsors' presentation. Ladies and gentlemen, after this we will have parallel session, but before that we will have a 60 minutes of lunch break. The lunch is held at the same restaurant as yesterday, so to all participants, speakers, please enjoy the lunch provided by the committee and the hotel on the first floor at Sanur Harum restaurant. And then at 1 p.m. we will start the parallel session. Please refer to the program book provided by the committee to see whether you are offline or you are online, and to see which room that you are registered into. To the online participants, after this at 1 p.m. we will start the parallel session, and please refer to the program book provided by the committee. At 1 p.m. our committee will provide breakout rooms, and please join the room that you are registered. And then to our on-site participants, for the offline participants, if after this you want to go directly to the room for your parallel room, this is the announcements. For room number 1, for the offline participants in room Gianyar, in Gianyar room, so all rooms are in this floor. So room number 1 is in Gianyar room, and room number 2 is in Amlapura room. Room number 3 is in Singaraja room, and room number 4 is in Mangupura room. Once again, for room number 1, Gianyar, room number 2, Amlapura, room number 3, Singaraja, and room number 4, Mangupura. If you have any information regarding the parallel session and the agenda, do not hesitate to come to forward. We will guide you. Thank you very much. And but please, right now, enjoy the lunch provided by the committee on the first floor at the Sanur Harum restaurant on the first floor, so we all can be energized back to continue the agenda for today's event. Thank you very much.
When the genetic code of our DNA was discovered, our world changed. We had identified the biological blueprint that defines everything about who we are. From there, the technology to explore our genome evolved rapidly. Programmable DNA editors like zinc finger nucleases and talents could change specific genes, and RNA interference could prevent gene expression. These tools transformed us from genome explorers to genome engineers. We could not only observe, but also change genes and investigate their impacts to understand how our biological blueprint is read. The advent of CRISPR-Cas9 technology brought versatility, expanding the engineer's toolbox to include virtually any type of change, from gene silencing to custom sequence insertion to transient activation or inhibition. Anything is possible. The efficiency of these methods allows us to explore and engineer on previously unimaginable scales, studying entire genomes
to participants.
Genome engineering has even entered the clinic, initiating an exciting new era of personalized medicine. CRISPR can
to participants and presenters both online and offline, please return to the main room after your parallel session. After all parallel session finish, please return to the main room. For the offline participants, for the online participants and for the offline participants, please return to this room as we will do a closing ceremony. Thank you. For now, please enjoy the lunch at the restaurants, and then if you have information, an announcement for the offline participants. If you are registered to room number 1, it is in Ruang Bangli, so Bangli room, not Gianya room. And then for room number 2 is Amlapura, room number 3 Singaraja, and room number 4 Mangupura. Thank you.
Apa kabar? Aku Intan dan aku akan membawakan kabar terbaru dari Merck. Merck sebagai prinsipal kini sudah terdaftar dan hadir loh sebagai penyedia dalam e-katalog LKPP. Nah, Sobat Merck bisa menemukan produk kami di etalase nasional untuk katalog peralatan laboratorium dan etalase lokal untuk katalog bahan kimia. Wah, sebuah kabar yang menggembirakan. Benar kan Sobat Merck? Yuk, kunjungi website e-katalog LKPP dan mulai berbelanja sekarang. Tapi gimana ya cara untuk temukan produk apa saja sih yang ada di e-katalog LKPP? Ingin tahu kan gimana caranya? Stay tune ya. Bye!
Kami percaya bahwa setiap hasil yang dikeluarkan haruslah sebuah aksi terbaik dari tindakan melayani sepenuh hati. Memberikan pelayanan terbaik dari hati, menjawab tantangan dan kebutuhan, agar Anda dapat menemukan pilihan sesuai kebutuhan Anda. Kami merupakan distributor alat laboratorium dan farmasi yang dapat terus diandalkan sejak tahun 1978. Fokus kami adalah untuk membangun Indonesia lebih maju lagi dengan memanfaatkan teknologi berkualitas tinggi di bidang laboratorium dan farmasi. PT Hartek Indonesia selalu berdedikasi membukakan jalan bagi teknologi terbaik dunia agar dapat diterapkan dengan maksimal sesuai kebutuhan Anda. Dengan pengalaman dan ilmu yang kompeten, PT Hartek Indonesia telah berkontribusi memajukan industri hingga universitas-universitas di Indonesia. Bersama dengan brand terkemuka di dunia, kami berkolaborasi mempersembahkan yang terbaik untuk Anda.